Hello, my name is Mia. I'm based at the University of Edinburgh in Scotland, and today I'm going to be talking about collaborations, offshore energy industry collaborations to improve the knowledge of elasmobranchs. So the aim of this study was to collate data on the occurrence and distribution of elasmobranchs associated with offshore energy industries to uncover new information on their populations and potential interactions with these offshore structures. So this study focused on the area in the central and northern North Sea and to the west of the Shetland Islands in Scotland. The coloured polygons in this map represent different offshore energy sites, so that includes offshore wind, oil and gas structures, wave and tidal test sites. So this is an overview of everywhere that we have collated data from. There's quite a lot of different data sources and streams, so for the purpose of this talk, I'm just going to be focusing on data that we access directly from industry themselves. So this has comprised mostly of existing survey operations and routine maintenance surveys, and from a variety of different methods, including ROV footage, commercial diver footage, and also surface sightings. I'm just going to focus on an example of ROV footage for today. And so this was taken from an oil and gas structure in the central North Sea to the east of Aberdeen. And this is a school of saith that are aggregating around this structure. And this was taken at about 85 meters depth. And shortly we'll see quite a nice image of a poor beagle shark coming into frame. And so from this type of footage, we can extract metadata such as date, time, depth, location, sex of the individual where possible, and also record any observations of behavior. Obviously, this is just a quick example, but throughout the study, I've reviewed over 360 hours of ROV footage. So I'm still processing some of the results, but I wanted to share some kind of key take-home messages at this stage. And so I think one of the most important things that this data is showing is that it's really filling key spatial gaps into areas that are typically inaccessible to traditional survey methods. For example, trawl-based fishery surveys, which are quite typical to this region. Kind of on the same vein, we're also able to gain some insights into, because these areas might be acting as natural exclusion zones or de facto MPAs, we're able to gain insights into how these zones might be supporting elasmobranchs through this function. We're also able to gain some quite unique behavioural observations from ROV footage in situ. For example, the slide previously showed an example of a poor beagle that might be displaying foraging behaviour, where they're potentially hunting for schooling fish that are aggregating around such structures. And lastly, but most importantly, it's really also highlighted the value of industry collaboration to enhance data coverage whilst using data that is already existing. So that's all I have time for right now, but please do ask me questions if you'd like to find more out about the results so far and the project at a wider scale. Thank you.
Thank you. We still have like 1 minute left for a question, if like anybody want to ask a question. Yes.
Thank you. Hi, Mia. Thanks for a great talk. I also work on offshore renewable energy, and I was just wondering how easy you've found it in the collaboration with industry to get them to give you data.
Yeah, that's a good question. So it has varied quite a lot across different industries as well. Through existing partnerships in my department, we've had really good collaboration with oil and gas industries. Slightly different results with the renewables, but we've still managed to gain access to a lot of their data. So yeah, it has kind of just varied case by case, but it's been good overall.
Okay, thank you.
Thank you.
Thank you, Mia. We're gonna go to for the next speakers. It's gonna be Jaime. He's gonna talk about 13 years elasmobranch stranding in Spain. Please, Jaime.
Hello, good morning. My name is Jaime Penadés. I come from Spain. I'm a researcher based in the Western Mediterranean. I'm going to be talking today about 13 years of strandings network working. Obviously, this is a very collaborative work with a lot of authors, but also a lot of students and anonymous people who helped along the way. I want to highlight the work of my colleague, Pablo García Salinas, because it was the first person where I did a necropsy. We were the ones that started all this project in the context of a marine mammal and marine turtle stranding networks. This is a common story in all over the world because marine mammals and marine turtles stranding networks are very well established, but for elasmobranchs, the story is quite different. There is an underdeveloped field. The causes for the strandings are multifactorial and often undetermined. Working with shark strandings, it's a very good opportunity for having full access to the individuals, for early career research training, and also for putting elasmobranchs on a spotlight, not only for conservation and research, but also for the local government and the general public. And it's a great opportunity for having studies that we could not have any other way. In Spain, for 30 years, we had this stranding network for marine mammals and marine turtles through the emergency call service, worked through the regional government, the university, and the aquarium. It had many instances of shark and race strandings, but they were overlooked, not attended, so that's why we started with this work. In the past 13 years, we had 65 incidents of 9 different species, 5 bathoid species, 4 sharks. The South region was the one with the most events, almost half of it because of the conditions of the sea. The most stranded species was the blue sharks, and the crazy thing about them was that 14 of the 21 blue sharks were stranded because of lethal interactions with swordfish. We have published a lot of things about this, but being stabbed by a swordfish is the second cause for elasmobranch stranding in our region, which is quite crazy. Okay, another mystery was the mobulus strandings. In the past year, we had 15 incidents, 6 of them for alive and they're left. This was common in the Western Mediterranean region. There were over 50 cases. We are still studying this. We are still writing an article, so we have nothing to comment on that. Thank you for these 3 minutes of your life. I hope it gets you many questions. And please say hi if you want or send me an email. And I think we have time for one question, maybe.
Thank you. Oh, okay, we have time for some question. 1 or 2, if that was. Oh, yeah, there's one.
Hello. So have you integrated the kind of alerts for elasmobranch strandings with the marine turtle and marine mammal strandings? So is it the same system that kind of records them?
Yeah, calls get through the emergency system, so everyone who's calling 112, they can say. It's very paradoxical because some of these species are more endangered than marine mammals and marine turtles. It's the same problem for society because it's a big carcass pottered in the beach and you have to retrieve it. So it was very easy to integrate, and I think this is something that you can do in your regions internationally to call your stranding networks dedicated to marine mammals and marine turtles and start getting that work for elasmobranch all over the world.
Okay, thank you. Thank you so much. We're gonna go to another speaker, Arzucan Askin. She's gonna talk about developing a community-driven national shark monitoring program.
Hello everyone, my name is Suzu, or at least that's how most people know me. I work in the Maldives, which is the only nation in the Indian Ocean that doesn't have a national targeted shark fishery. It's 99% ocean, so most of it is actually water. So it's a place that has a lot of incredible habitats for sharks, but we know very little about the 40+ shark species that inhabit our nation. And what we want to find out more is, how does habitat loss, climate change, and pollution affect them? Because those are the biggest threats that we have in country. So why citizen science? As a researcher, I can't possibly survey a country that consists of 1,200 islands and spans 800 kilometers alone. And millions of people come to the Maldives every single year, and thousands work in the diving industry. So we can mobilize this whole community of ocean users to collect the data together with us. And what happens when we do that? We get a lot of interesting information about some of the most exciting things that we're looking to uncover about the sharks. So we launched the Maldives Shark Hub, which is our national platform for citizen scientists and researchers to collect sightings. It has lots of targeted user groups. Oh, you scared me there with the 1 minute left. So it's specifically designed for dive guides, tourists, and fishers who have co-designed this hub with us. Here's a bit what it looks like. It's much scarier on screen. Go download it on your phones and actually take a look. We collect a variety of observational data through the Shark Hub, everything from shark sightings and IDs to really interesting ecological events. The forms are quite simple. They've also been refined by our users. And what's amazing is that through this, we're getting a lot of incredible observations, like, for example, this whale carcass in North Ari Atoll, a lot of interesting and very exciting first ever observations of mating behavior in juveniles, which we're actively looking for to identify those key areas, and photo IDs. So we do photo ID using multiple different methods to identify individual sharks. We do this for oceanic white-tip sharks across the country, great hammerheads, bottlenose wedgefish, bowmouth guitarfish, leopard sharks, and also tiger sharks. When users send us an ID, we get a notification. Then we can go on the back end and review the IDs. Once we've gone through it and confirmed if it's a new or known individual, the user automatically gets notified. They also get an ID card. In this case, the first guitarfish that Ricky, our president, entered into the database ended up actually getting named by Dr. Sylvia Earle. And we can also track all of the sightings of one individual in one place all across time. So every single photo ever taken of that shark, every single sighting submitted, all that metadata is available in just one click. We also get really unique observations of individuals that we otherwise wouldn't be able to see so often and can track their mortality and survival over time. When people note a deceased shark in the app, our team gets a notification and we can go and retrieve the specimen. And some of the specimens ended up in our collection, and one of them was the first ever shark specimen dissected at Maldives National University. The user experience is easy. Everybody has a dashboard, so you can track everything you've submitted over time, and it adds points. And at the end of the year, we have a Shark Hub Wrapped, a bit like Spotify Wrapped, to keep it exciting. Thank you.
Thank you. Any other question? Oh, seems you have a lot of fans. Any other question? We still have 1 minute 30 seconds. Okay, thank you. That is all published report.
Thank you very much. So let's start immediately. For those of you which are not familiar with Mediterranean, Mediterranean is a semi-closed sea connected to Atlantic with the Strait of Gibraltar and also to Indian Ocean through the artificial channel Suez Canal. On the north we have a European continent, on the east we have an Asian continent, and on the south we have an African continent. So in recent years I found joy in the myth-busting business. What does it mean? That there are some myths or traditional beliefs that we found essentially wrong and we are trying to correct them. For example, one of the strongest traditional beliefs is that the species richness of the sharks, rays, and chimeras is the highest in the northern Mediterranean and western Mediterranean. And we, being a regional vice chair of IUCN Shark Specialist Group, I have the pleasure to communicate with lots of people from the eastern side and the southern side, and we found it that essentially wrong because we know what is the basis of it. And the basis of it is that in this area, which means western and the northern area, that everybody speaks pretty much English, and all the reports are written in English or in European languages, which are later translated into the English. So it's pretty much easy to get information from that area, and that's the reason why this area is very well studied. And most of the information are arriving from these areas. However, what about African side and Asian side? For many years and many decades, actually, these information that we are getting from these areas were scarce. Why? Because there are some information that have been published in other languages than European languages. For example, in the eastern area, they are publishing lots of papers in Turkish, also published in local journals that they were using only Turkish language. Also in Arabian countries, of course, even the shark research in Arabian countries was not so developed because, you know, they have different priorities. And this is the reason why, at the end, Northern and Western Mediterranean were considered as the areas with the highest species richness. But we tried to include all kinds of the reports from all the areas, not only research articles, but also different kind of reports in different languages, and we found out that actually the situation is completely different. That the Eastern Mediterranean is more diverse in terms of species richness than Northern and Western Mediterranean. And unfortunately, we still have some areas, for example, southern side, shall we say Libya, where we cannot get more information. But this changes this traditional belief, and right now we think, and very soon we will write a paper about it, that Eastern Mediterranean has more species richness than the western side and northern side. So this is the way how we are myth-busting these traditional beliefs, and hopefully in the future this belief will be changed correctly. So this is pretty much it, and thank you.
Thank you. We still have time for one question. Any question? Yep.
Hi, Alen.
Hi.
Thank you for this. This is amazing work, and of course, I agree. Do you think now with the AI it will be, the reports from the eastern side will be more taken into consideration with the new data?
Well, hopefully, as you know, 2 decades or 3 decades ago, we didn't have any kind of information because essentially the research in those areas were not very, very established. But right now, we are trying to communicate with all of these people and trying to involve that, trying to force them that these reports should be published in English, in hopefully, I think, for example, in Turkish case, now they are publishing a lot of papers in English-speaking journals. So I believe that we will have more and more information from these areas, and hopefully at least from the South Mediterranean, where this is a sort of a vast desert for information.
Thank you so much. We're running out of time. Carly?
So when I put this presentation together, I was actually worried about how to do justice to 15 years of research in a 3-minute presentation, especially considering the number of publications, projects, and students involved. It actually worked out that I'm going to have about 2 seconds per publication and about 12 seconds per year. So get ready for it. And the best way that I came about to do this would be to do some kind of quick fire Wednesday about all the pros and cons that are typically mentioned when we talk about wildlife tourism or shark tourism, using the wild shark cage diving industry, which is what we're working on mostly. One of the main things that people typically refer to is education or conservation awareness, and we tested this by doing some survey before and after the experience, and we've shown that these participants have actually increased their conservation behavior as a result of the cage diving experience by donating, being more likely to donate, but also talking to friends about conservation actions as well. We also estimated the value of shark tourism in Australia at 26 million, and in terms of the cage diving industry, there's more than 85% of the people that would not have come to the region, and even international tourists would not have come to Australia if it wasn't for the cage diving industry. Cage diving is also, in wildlife tourism, is also a great platform for research. It facilitates a huge program on
What's that? Like, I don't know.
That fire alarm, do you want me to continue?
Okay.
I'll continue if you want.
Keep going. No, keep going. Keep going.
It facilitated the research in terms of shark bite mitigation measures, but also in terms of pure science. And we've deployed now some temperature sensors on wild sharks that tells us about more of their metabolic rate. We've also, obviously, we know that wildlife tourism can also have some negative impact, and behavioral impact is something that is often referred to. So we looked at the residency of white sharks, and there was indeed an increased residency as a result of an increase of intensity of cage diving, but through mitigation measures and new regulation, we're able to show that the residency went back down to baseline levels and has done so for the last about 14 years. There's also some concerns about individual fitness of these individuals through maybe increased energetic expenditure when they interact with the cage diving boat. And whilst we have shown that there is indeed an increase of burst events, which is energetically costly, these burst events occur at such a low frequency in short duration that when applied across a daily energy budget, it only led to an increase of less than 1%. In South Australia, we also have community concerns about human safety and the potential association that might be occurring or conditioning with white sharks learning to associate food and humans or boat. So we looked at that from the perspective of how long do white sharks spend along this boat and whether they increase or decrease. And what we found is like the opposite to what we expected, with a decreased amount of time in proximity to the boat, likely to be more a case of habituation with a loss of response rather than the conditioning that people were worried about. We're not just only worried about the white sharks, we're also looking at the whole of the ecosystem and other species. We found an increase in abundance and residency for a couple of the species, but these species were still undertaking the natural migrations. And when we looked at the entire ecosystem in the fish community, we showed that the Neptune Islands is actually no different to some of the other offshore nearby islands as well. So once you put all of that together, we use kind of a novel multidisciplinary comprehensive framework using some of these factors here to try to look at the sustainability of that tourism overall, and found that all of the research is now being used and implemented in the management and the policies of the cage diving industry. And that's also thanks to this kind of holy trilogy of the collaboration between government, scientists, and industry. But realistically, we also need to take account community. And it's not really a trilogy, but now we're referring to more of a holy tetralogy. Thank you.
Thank you so much. Is there any question? We still have time for one question for Charlie. Yeah.
Hello, thank you so much for the presentation. I was curious, what kind of measurements did you apply to get back the residency levels of that shark species that you mentioned? After new regulations, they got back to their normal residency patterns?
You're talking about the regulations?
Yes.
Yeah. They have now a penalty so to try to limit how much burleying and baiting they're using. And they also have a limited number of days that they can be on the island. And we're able to modify that number of days depending on residency. If residency increase, they're more limited of how many days they can be there. If residency goes back down, they can go back to more days.
And you have, the area have ways to regulate that so that people follow the instructions?
Yeah, there's some really good relationship between industry and manager, and we can ensure compliance of that as well.
Amazing.
Thank you so much. Please give applause to all of our wonderful speakers. Okay, next session gonna be Brynn Devine. She gonna talk about satellite telemetry of Greenland sharks. Okay, enjoy the talk.
Thank you. Hi everyone, thanks for coming. So today I'm going to talk about seasonal movements of Greenland shark, and this is some work that I did as part of my postdoc with Nigel Hussey at the University of Windsor in Canada. I'm going to stand this way so I can see my slides because I'm so short. So I'll just start by introducing the Greenland shark for those of you who may not be overly familiar with this species. So Greenland sharks have a widespread distribution throughout the North Atlantic and adjacent Arctic waters. Because they are so widespread, they do interact with a lot of different northern fisheries. So there's some targeted fishing in places like Iceland and Greenland, but most of it is incidental bycatch. So they are accidentally caught in several trawl, longline, and gillnet fisheries. We do know these sharks have some rather extreme life history traits. Based on recent radiocarbon dating of the islands, they're now believed to be among the longest living vertebrates with lifespans estimated to exceed 250 years, with potential age at maturity of 150 years. And this combination of having these sensitive life traits and being a common bycatch species, they are considered to be vulnerable by the IUCN Red List. So now jumping into their movement, studies have aimed at addressing some of the outstanding knowledge gaps related to this species. And this includes identifying the location of both mating and pupping grounds, which are currently unknown. Satellite telemetry can be a very useful tool, but as many of you know, can be quite challenging for deep water species like Greenland shark. Previous studies have revealed, however, that Greenland sharks, despite being sleeper sharks with very slow swimming speeds, they are capable of making very long distance movements. But most of the tag deployments to date have been between 3 to 6 months, so it's still a long time, but we may be missing some seasonal variations in their movement. And to sort of confirm this, there have been seasonal differences in bycatch rates observed between regions, so there's really a need for more information on their seasonal movements, and this will be necessary in order to better inform mitigation strategies to reduce bycatch. So we wanted to know, would longer tag deployments detect potential seasonal movement patterns for Greenland shark? So to answer this question, Greenland sharks were caught using long lines in 5 locations in the high Arctic in Nunavut, Canada. This included 3 fjords on Baffin Island and 2 fjords near the communities of Resolute and Grise Fjord, and this work was done in late summer. So each shark was measured, sexed, and then tagged with a mini-pat, a satellite tag that was programmed to stay on the shark for 1 year. And in a hope of providing some additional geolocation points between that tagging and pop-off location, because they don't come to the surface very often, each shark was also tagged with an acoustic tag, as well as a subset of 7 sharks tagged with 1 or more MR-PAT satellite tags that were programmed to stagger release. So a total of 14 Greenland sharks were tagged. They ranged in size from 150 to 322 centimeters total length, and an equal number of male and females. So 11 of these sharks provided data for at least 358 days. 3 tags did release early, 1 unfortunately after just 80 days, but the other 2 still reported data for 280 days. So we got some really long deployments, which is great. So this map is the same map as the previous slide. I've just added the mini-pat pop-off locations. So you can see, and they're color-coded by the tagging locations. So you can see roughly 1 year later, some sharks were very close to where they were captured and tagged. Others not so much. They maybe went to other inshore fjords in Nunavut and Baffin Island. Some went to Greenland. So we do know that most of the sharks did return to Baffin Island, and you can see most of them did return to the high Arctic in these inshore areas in Nunavut. And so looking at the depth and temperature data for all of the sharks, so this is all the data pooled for all individuals, over 90% of the deployment time they spent at mesopelagic depths between 200 and 500 meters. And then in terms of temperature, so not looking at seasonality, less than 5% of the deployment time was spent in sub-zero temperatures, but about half of that time was spent in relatively warm waters between 5 to 6 degrees Celsius. So now looking at the seasonal variations, we obviously wanted to dig a bit deeper into the depth and temperature use throughout the deployment period. So I have a variety of plots here. We'll start on the bottom left. So you can see that this is just a classic example of one individual for depth and temperature use over time. You can see there's a really marked difference in temperature use during the middle of the deployment period. And this corresponds to winter and spring months. So moving to the right, these are all of the individuals. Looking at the top left first is mean monthly depth use. So you can see the depth was relatively stable throughout the deployment period for most individuals. There was one weirdo doing I don't even know what in the deep sea. But for the rest of the individuals, pretty consistent, but especially compared to temperature. So that's the top right, and then the larger panel below is the mean monthly temperature use. So you can see sharks are accessing those warmer waters above 5 degrees starting in December, January, and then going through April, May, with a few individuals staying a bit longer in these warmer waters. So then the big question, where are these sharks finding plus 5 degrees Celsius water in the Arctic in wintertime? That's pretty warm. So this is an example of one of those sharks, and again, including some of those additional geolocation points from the MR-PAT tags and the acoustic tag. So you can see the shark was tagged in August in the high Arctic, northern Baffin Island. The staggered MR-PAT releases show that the shark exited the fjord and went a bit north into Lancaster Sound, which is at the opening of the Northwest Passage, before apparently moving south, as the shark was then detected in January in Davis Strait, so that's that narrow portion between Greenland and Canada. We know from the tag data that the shark was then in warmer waters, and then it was detected again in June just inside Davis Strait, and then the mini-pat eventually popped off pretty close to where it was tagged. So we can see that these sharks are finding these temperatures by exiting Baffin Bay and and leaving the Arctic entirely. So to do this a bit more sophisticated, we called on some collaborators, Martin and Cameron Braun at Woods Hole Institute, to do some geolocation modeling. So the model output shown here for each of the seasons suggests that there is this potential north-south movement of Greenland sharks. So they are tagged, or at least this subset of sharks, tagged inshore in the summer in Nunavut, and then transiting through Baffin Bay south, and then accessing these warmer waters in the Labrador Sea in winter and spring. So in terms of next step, just really quickly, the Labrador Sea seems to be an important region for Greenland sharks, but there's been no prior tagging efforts. So to address this gap, we've started a tagging program in Nunatsiavut, which is a region in northern Labrador. My coworker, Sid Payne, has lived there for over 30 years, and we've been partnering with the Nunatsiavut government to do some scientific longlining to try to tag sharks ourselves, and we're also partnering with governments in both Canada and Greenland to opportunistically tag Greenland sharks caught in their trawl surveys. So we've tagged 7 sharks so far. We hope to add some more this year. Our preliminary results do support the evidence of this being an important area for Greenland sharks, but of course these sharks are doing something a little bit different, so we really need some more tagging to hopefully answer some of the more questions that we have. So just to give some key takeaways, the longer tag deployments did show a potential seasonal north-south movement for this species, at least in the sharks that we looked at. So these are sharks caught inshore in the summer in Nunavut. And we also provided further evidence of long-distance movements for these slow-swimming sleeper sharks. So a round-trip journey from the high Arctic to the Labrador Sea and back is well over 2,000 kilometers, so they're really moving quite far. The Labrador Sea does seem to be an important overwintering area for some sharks, with clear connectivity with Greenland, which isn't surprising for the Greenland shark, but it does highlight the need for strong cross-country collaboration in terms of management and conservation strategies that are both collaborative and complementary. In terms of what's driving these seasonal movements, they could be driven by ice dynamics or prey distributions. We're not really sure, but there are clear implications for fisheries bycatch, and particularly in the summer and fall, as that seems to be the time where there's a lot of offshore fishing happening in Baffin Bay, but it's the same time that these sharks might be transiting. So it could be helpful to have season-specific mitigation measures that could be helpful to reduce shark encounters. And with that, I'll say thank you and happy to take any questions.
Thank you so much. We still have very short time for questions. Is there any question about it? Yes, over there. Maybe.
Hello, Patty Burke here. Really cool talk and very exciting to see that you put out 14 tags and you actually had 11 report. That is huge.
Yeah.
Congratulations. I was actually curious, how did you actually go about tagging it? Was it that last picture that we saw, like the dorsal fin mount?
Yeah, similar to that. I should clarify, I was not present for the field work of these tags, but similar. They're scientific long lines, so typically a soak time 5 to 6 hours, maybe 10 hooks, and then, yeah, kind of taking the sharks when they come to the surface to a smaller vessel to do the tagging. I'm not sure if that answered your question, but I can direct you to others who were there for the field work.
Thank you. Okay, thank you. Very great talk.
Good morning. Thank you all so much for coming. My name's Emma DeBow from the University of Newcastle, Australia, if the accent wasn't already painfully obvious enough. And I am so thrilled to be presenting to you my honours research examining the effects of ray bioturbation on macroinvertebrates and sediment characteristics. This work was conducted in 2025 and was supervised by Troy Gaston, Margaret Patel, Vincent Rout, and Molly Grew. Bioturbation refers to the reworking and disturbance of sediments by organisms. Ray bioturbation is conducted to extract infaunal prey buried within the sediment and is done using the hydraulic jetting from the mouth and spiracles combined with coordinated pectoral fin movements, as demonstrated by this estuary stingray. This process results in the formation of depressions in the sand known as ray feeding pits. Ray feeding pits are circular, with 2 small parallel mounds of sand on either side of the pit. They're frequently observed during low tide and have been recorded to last up to 14 tidal cycles, with depths ranging from 1.5 to 10 centimetres deep. Despite the prevalence of feeding pits, minimal research has examined the effects of ray bioturbation. and those that have either focus on sediment alone or macroinvertebrates alone. One such study by Molly Grew in 2024 found that within a 24-hour period in an estuary, rays moved the amount of sand at a weight equivalent to 16 Sri Lankan elephants. That is a huge amount of sand being moved, but we don't really know the effects this has on the environment. Similarly, the effects of macroinvertebrate bioturbation have been well recorded, of having impacts on biogeochemical cycling and other macroinvertebrate behaviour. So, to address this gap, my research aimed to investigate the relationship between ray bioturbation, sediment, and macroinvertebrate communities across 2 wave-dominated barrier estuaries along southeastern Australia. The estuaries are approximately 300 kilometres apart, with Behavioural Lake being an intermittently open system where sand can influence tidal exchange, whereas Brisbane water is permanently open and more tidally flushed. To isolate the effects of ray bioturbation, I collected 2 samples. So 10 ray feeding pits were visually identified and samples were collected from them. And then 1.5 metres away from each pit, I collected samples as well. I collected both sediment and macroinvertebrate samples. So sediment samples were collected using 30 millilitre cores, and the oxic-anoxic interface, which is the visual barrier between oxygenated sediments and anoxic sediments, was measured in situ. And later in analysis, the core was determined as either being predominantly oxic, predominantly anoxic, or indiscernible. And the samples were retained for later grain size analysis and organic matter content analysis. Macroinvertebrates were collected in 7 centimetre core samples, sieved through the field in 2 millimetre mesh, and later identified to the family level. And the sampling was conducted in 2 periods, March and August, with March in Australia being warm, about 25 degrees average, and August being colder, 19 degrees average. So the Permanova results demonstrated that, contrary to expectations, grain size distribution was not significantly different between ray feeding pits and non-pits. And organic matter showed some site-specific variation, but overall the patterns were consistent. However, as demonstrated in this figure, pits consistently demonstrated an indiscernible oxic-anoxic interface compared to surrounding sediments. So this figure, in the orange on your left, you can see in August, non-pits were predominantly oxic, whereas ray feeding pits were consistently indiscernible, while in March, this pattern swapped and non-pits were anoxic, but pits stayed as indiscernible. This is likely because ray bioturbation acts on pre-established oxic-anoxic interfaces as an independent mediator. And using LLMs, the macroinvertebrate response varied over time as well. So in March sampling, abundance was higher in pits than non-pits, whereas in August, this flipped and abundance was higher in non-pits than pits. Family richness followed a similar pattern, but overall diversity remained unchanged. Additionally, certain taxa, such as nudibranchs, herbivorous gastropods, and predatory polychaetes, were found exclusively in pits, which suggests that the ray feeding pits may act as further trait-filtered microhabitats. So, overall, the patterns of macroinvertebrate distribution align with patterns in the oxic-anoxic interface. In March, as pit samples were predominantly categorised as anoxic, macroinvertebrates had an increased presence in pits compared to non-pits, which then reversed in August, when non-pit sediments were predominantly anoxic, indiscernible or oxic, macroinvertebrates increased presence in non-pits compared to pits. This suggests that under certain climatic conditions or oxygenation conditions, ray feeding pits may act as advantageous microhabitats that are beneficial to macroinvertebrate survival. However, the outcomes of this study are obviously limited by the reliance on the visual identification of the oxic-anoxic interface and limited sampling periods. However, these findings are particularly exciting because they are among the first to acknowledge there is an interrelationship between ray bioturbation, macroinvertebrates, and sediment properties. And it really emphasises the role of rays as ecosystem engineers. It opens avenues for future research, as there is potential for the relationship between ray bioturbation, the oxic-anoxic interface, and macroinvertebrates to become increasingly important, as ray feeding pits may become important reservoirs of water and dissolved oxygen under climate change. This has implications for estuary management and really reinforces the importance of conserving ray populations. I think I have some time for a few questions, but otherwise, please add me on LinkedIn or come have a chat. I'd love to talk about this more.
We do have time. We do definitely have time. We do, in fact, have time for questions. Any questions?
Hey, Emma, Patty here. Great talk, as always. Really cool research, and I think you're really highlighting a lot of the important work that I think sometimes people don't initially see right off the bat. So thank you for sharing your work. I was interested to think just maybe more of a question, more than general thought-provoking question. Yours was done in New South Wales, right, in the more temperate area. Is there any work that's been done in more tropical areas on this sort of work, as you know, temperature will affect that anoxic layer a little bit? So more of just a question, I guess, but thank you for your time.
Thank you. Yeah, of course. So a lot, the strongest body of work that has looked at all, like the interrelationship between all three, was done in 1984. And it did not look at oxygen at all. Oxygen, part of the reason why the oxic-anoxic interface was a visual indicator instead of actually measuring dissolved oxygen is because no one had looked at this before, at least not that I could find anywhere or published at the time of writing. And so it wasn't expected that it would influence oxygen dynamics at all, or this oxic-anoxic interface. So to my knowledge, no, but I agree it would be very interesting. And when we talk about Australia with the algal blooms, how it could have implications there. Yeah. Thank you for your question.
We still have time for one more question.
Excellent. Hello. Excellent presentation. What was the size range in the pits that you were using? Were they relatively similar in size,
yes.
Larger, smaller?
So even though I was really biased to the big, beautiful ones, I had a range of pits, and they ranged from, I believe, 2.5 centimetres in diameter to 7 centimetres in diameter. And just as a side comment on that, not my research, but other research has tried to look at the relationship between species and the formation of ray pits. And it looks like ray pit size isn't a reliable indicator of ray species. I know that's not what your question, but just when we talk about ray pit size and dimensions, that I would have liked to integrate that, but yeah. And I did actually do some testing. There was no relationship between ray pit size and macroinvertebrates or sediment.
Thank you very much, Emma.
Thank you, everyone.
Next up, we have Alex Hearn, and he will be presenting on the Paramount ISRA, a connectivity hub for critically endangered scalloped hammerhead sharks.
Good morning, everyone. What an amazing conference this has been so far, and we're only halfway through, 2.5 days. I have a notebook full of try this at home comments, and I've just graduated to sending try this at home comments to people who are asleep in Galapagos right now, so thank you for the previous talk. My talk was originally meant to be in the ISRA session, how to leverage important shark and ray areas, but I think it got moved because there has been such a great, how do you say it in English, acogida, acceptance of ISRAs, and we've kind of spilled over into all these other sessions too. So I'm going to assume that you already know about ISRAs, and if not, then definitely go to the session on Friday. Meanwhile, I've changed the title of my talk to make it fit in a little bit more to this session. I guess I'll start by saying it's probably given for most of us here, especially if you were at Marta Cambra's talk on Monday, that seamounts tend to attract wildlife, including sharks. And today I want to talk about a particular seamount and about how it attracts a particular shark species, the scalloped hammerhead shark. This is a critically endangered species that's showing declines across most of its range. Surveys at Cocos and Galapagos Islands in the eastern tropical Pacific suggest declines of around 40% over the last 2 decades, despite strict protections in place. Now, although there is some documented illegal shark fishing in both reserves, it is generally accepted that this is more to do with the sharks moving outside protected waters. And what is becoming clear is that simply protecting these hotspots or aggregation sites and carrying on business as usual outside is not enough. So here's the ISRAs. Here's a zoom in on the eastern tropical Pacific, and you can see our ISRAs here. You'll note there's a kind of a nested composition to those, and that's a discussion for another time. But I want to draw your attention to a tiny dot halfway between Galapagos and Cocos. That tiny dot, Paramount, is the ISRA that I'd like to talk about. So with a peak only 190 meters from the surface, Paramount is the only shallow water seamount along what we call the Cocos-Galapagos swimway. This is a Mission Blue Hope spot. It's highlighted in light blue here. And it's a loose chain of seamounts linking Cocos Island with Galapagos, and along which species of several different taxonomic groups move, including, of course, sharks. Now, if you look at the red outlines depicting the MPAs, you can see the Galapagos Marine Reserve down here, Cocos up there, and then we have this Hermandad Reserve, this new reserve that Ecuador created back in 2022. But you'll notice that Paramount is actually outside that reserve, about a stone's throw outside that reserve. When Ecuador undertook the process to create Hermandad in 2022, there were some data that pointed to the importance of Paramount, but it was excluded during the consensus building process with different stakeholders, mainly due to pressure from the industrial longline fleet. At the time, the data we had was fairly sparse. There'd been 2 expeditions there. On both occasions, we'd filmed large schools of hammerheads. And we had some evidence from satellite tracking that sharks tagged in Galapagos did utilize Paramount. That was pretty much it. We did suspect, though, that its role might be more important than we could demonstrate at the time. So rather than simply let it go, we resolved to explore the seamount further. The existing data, however, were used to justify its designation as a rather small ISRA a couple of years later. It's one of the first ones to be designated, but it basically covers a 12-kilometer radius around the peak of this seamount, centered on the shallowest peak. So the area is about 160 square kilometers. So this was important for 2 reasons. The fact that it was now recognized as an ISRA helped us to justify including it in the monitoring plan for Hermandad. The idea being that this is an area that could potentially benefit from the protection of Hermandad, which was designed as a migratory corridor. So it means that it's now one of the 19 permanent monitoring sites that we have to evaluate Hermandad. And this kind of keeps it on the radar for decision makers and stakeholders working in that region. Secondly, of course, the fact that it was an ISRA allowed us, my team and I, to leverage support from Mission Blue, Galapagos Conservation Trust, and Bezos Earth Fund to carry out an expedition in early 2025 to explore exactly how scalloped hammerheads were using the seamount. Got it. Okay. So during that expedition and a couple of earlier expeditions, we placed spot tags on 6 female and 10 male scalloped hammerheads, all adults. So this table is a little outdated now because some of the sharks are still checking in. But the average track length here is about 260 days and up to, including today, about 607. And you can see the tracks. Basically, we have animals that are moving widely across the area, but there's quite a lot of heavy traffic between Galapagos, Cocos, the swimway, coastal Panama. This is something that we knew, but we wanted to break it down by sex and start looking at how each sex is using these areas. And so what you can see here, this is just a graph that shows in darker, less percentage of use, lighter, higher percentage of use of different ISRAs. And we expected to see the bottom line. Most of these sharks are utilizing the Cocos Galapagos swimwear very heavily. But if you look at the top right, you can see the males in particular are hanging around Paramount Seamount. We've got males there that stayed for pretty much 365 days in a year. And if we pass that out, where were the females going? Well, the 4 females that lasted more than 200 days all traveled to the same bay in Panama, actually to another ISRA, which was created to protect pupping grounds for scalloped hammerheads. So this is, we had evidence of this in the past, but these are 4 females arriving within a week of each other, spending a couple of days there, turning around and coming back along the same path, again stopping by Paramount, but only very briefly. So it seems that Paramount is being utilized by each sex very differently. The males along the bottom here. On the left, you have areas of residency. On the right, you have areas of movement. The females don't really reside anywhere in terms of their behavior. They seem to be moving around much more, whereas the males are staying put. Now, there's another part of this story, which is, okay, so how do we go from this to protection? Well, there's several things. When Hermandad was created, we were asked to carry out an economic analysis of how the protection of Hermandad might affect the economy of fishermen who were fishing there before. And when we analyzed this, the fishermen asked us to already include Paramount in these analyses. They felt that they were unable to set their long lines, even though it was unprotected, because they were worried they would drift into the new marine reserve. So this economic study formed the basis as a package of assistance to fishermen in exchange for the creation of Hermandad. So the take-home message here is that any potential loss of income resulting from excluding fishing from Paramount has already been considered and accounted for, even though there's no protection in place. So where do we go from here? 2026 is an important year for seamounts, with the adoption of the Conserving Seamounts proposal at the Convention of Highly Migratory Species earlier this year. By including Paramount within the Hermandad monitoring framework, we've kept it on the government's radar. We're letting the data from the monitoring speak for itself. So technical staff from the park, the Navy, the Fisheries Institute are able to see what the data is creating. We already have senior staff at the Galapagos National Park expressing the importance of Paramount and the option of including it in an expansion of Hermandad. And finally, we are working on generating more awareness nationally about the importance of this site with a documentary coming out later this year. So we're going to have a busy year switching from biology to policy and sitting down with the stakeholders and hopefully getting this included in the new reserve. That was pretty quick, but I'd just like to thank the organizations who have supported us. And if you have any questions, I'll take them now or over coffee.
Over coffee. Thank you very much,
coffee.
Alex.
All right.
Next up, we have Sasha I Faul, and she's been speaking about training nurseries for exploration. Tiger sharks employ a novel life history strategy across ontology.
All right, hi everyone. Thank you so much for coming today. I'm really excited to talk to you about the 3rd chapter of my master's thesis about juvenile tiger sharks on the east coast of Australia. So movement and habitat use by tiger sharks is relatively well studied, and we see a lot of different examples of strategies such as nomadic behaviors, resident behaviors, or transoceanic migrations, seasonal migrations, and so on. But all of this tracking data, all this information comes predominantly from sub-adult or adult tiger sharks. We really don't know much about what the small juveniles are doing, and in particular, young of the year, or sharks less than a year old. And this presents quite a significant knowledge gap. For such a cosmopolitan, well-studied species, we really don't know much about what these small juveniles are doing. So for example, do they use strategies similar to adults? Do they use distinct essential habitats? And does their habitat use change across ontogeny? So to address some of these questions, we used acoustic telemetry. And Australia has quite an incredible acoustic array on the east coast. All the pink dots on that map there are the locations of acoustic receivers. And this is facilitated through IMOS, Integrated Marine Observing System. Now, obviously, it's not comprehensive cover of movements, but the scale of it allows us to get some pretty cool pictures into the large-scale movements. And finally, given the really broad distribution of tiger sharks on the East Coast, we split the data into two regions. So the north, the Coral Sea Marine region, and the south, the Temperate East Marine region. And the northern region tends to be a lot more tropical, similar climate to here in certain areas, whereas the south is cooler and experiences more dramatic temperature shifts. So we split the tiger sharks that we tagged into different size classes based on their total length, from the tip of the snout to the tip of the tail. And the ones on the right, so the neonates, the young of the year, the small juveniles, these are the really poorly studied life stages. Whereas our large juveniles, our subadults, and our adults, we know a little bit more about what they're doing. So sharks less than about 205 centimeters, these are the really poorly studied life stages. So these are the tagging and detection locations of sharks in this study, where the red cross represents tag location and the colored dot is a detection location. Now, we wanted to include the best picture of what sharks were doing, so we only included sharks for analysis which had at least 100 days of tracking. And unfortunately, a lot of our young of the year were no longer young of the year by the time we got good data from them, so we only had 2 that we could include in the analysis. However, we did have 60 small juveniles, which is quite an incredible sample size for this poorly studied life stage. Then we had 147 large juveniles and 196 subadult tiger sharks. But an important consideration is some of these tiger sharks move through different size classes across the tracking period. So using growth rates from the east coast of Australia, from Holmes et al., 2015, we actually assigned an age to the sharks based on their length at tagging and subsequently estimated how old they were when they were detected. And this is a really important consideration for long-term tracking data. So the first thing that we looked at was maximum distance from the tag location. So that's the single largest distance. And on the y-axis, you can see the distance in kilometers, and on the x-axis is the age of the shark in years. And this is for sharks tagged in the north. We saw a general trend of increasing maximum distance with age. But flagging that sample sizes were quite low for the young of the year and the small juvenile life stages. However, when we looked at sharks tagged in the south, we saw no trend in maximum distance against age. And incredibly, 2-year-old sharks had the highest maximum distance, on average at 750 kilometers, which is quite incredible. Another important thing to note is that the scale of movement was really different in the north versus the south, where in the north, sharks were generally moving not as far distances, a little bit smaller than in the south. But I'll get to that a little bit later. So the next thing that we looked at was residency index, which is on a scale of 0 to 1, calculated as the number of days detected close to the tagging location divided by the number of days monitored for. And there was no trend in residency index against age in either the north or the south, and it was quite variable across different ages. But again, interestingly, age 2-year-old sharks had the lowest average residency in the south. And you can see that the scale of residency was quite different in both the north and the south. Sharks in the north were generally much more resident than in the south. Now, if we think back to that division between the northern region and the southern region, overall in the north, we've seen lower maximum distance and higher residency index, while in the south, higher maximum distance and lower residency index. And this is likely due to the environmental differences between these regions, the environmental context, or temperature. And this is relatively well known for tiger sharks on the east coast of Australia, that temperature does indeed drive their movements. However, it was really interesting to see the effect that this has on all life stages, including those poorly studied small juvenile tiger sharks. Now, I mentioned that we don't really know much about young of the year, so I wanted to include the tracks of some young of the year just to give a bit of a picture of what they might be doing. So here are 2 that were tagged in the north, both of them relatively close to where they were tagged around the island groups. But here are 2 that were tagged in the south. Both of them, in a matter of days, swam close to or over 100 kilometers. Now, this is obviously very much preliminary information, but it's quite impressive to see this capability or capacity of young of the year to make some large-scale, relatively large-scale movements. Now, there's definitely a need for more tracking data on young of the year, but so far we're seeing highly variable movement, similar to adults. We're seeing this variability partially driven by the environmental context, that small juveniles frequently make large-scale movements, and that they exhibit low residency. And these two in particular seem a little bit unusual in the elasmobranch world, where generally large-bodied sharks, such as lemon sharks or bull sharks, scalloped hammerheads, they use these discrete nursery areas, these sheltered areas, that meet these three criteria of high density, extended residency, and repeated use across multiple years. But with this data from the small juvenile tiger sharks, we haven't actually seen that they've met these criteria, especially in the south where there is low residency and high mobility. So if they're not using nurseries, what could they be doing instead? Well, when we compare with some other species, such as gray seals, emperor penguins, or albatross, juveniles of these species tend to do exploratory strategies of habitat use, where recently fledged or recently weaned animals depart the colonies and make these really large-scale movements while they learn how to forage, learn how to find habitats, and survive. It's also possible that the movements we've seen are a reflection of the nomadic and migratory strategies used by adult tiger sharks. So, while our preliminary findings support these potential exploratory or migratory strategies, there is absolutely a need to do more research into juvenile tiger sharks, especially setting out to test the three criteria, explicitly test the three criteria, and with a focus on young of the year tiger sharks. And with that, thank you everyone. Thank you to my co-authors and supervisors, the Biopixel Lab, and especially all the funding that made this possible. Thank you.
Thank you so much, Sasha. Thank you so much for your talk. Sadly, we have no time for questions. Up next, we will have Hendrik Winter, who will be speaking about offshore wind, electromagnetic fields, and benthic elasmobranchs.
Okay, hello everyone. I'm going to give this talk on behalf of Annemiek Hermans, who at the last moment unfortunately could not attend this wonderful symposium. She feels very sorry about that, but I'm one of her supervisors and co-worked on all her work, so I'm stepping in for her, and I hope to do her work justice. I'm going to tell you about offshore wind, electromagnetic fields, and controlled experiments we did to see what effect they have on benthic elasmobranchs. So the offshore wind energy is a rapidly increasing industry, and the North Sea is one of the front runners in that worldwide. There's a lot of sites where new wind farms are being built, and there's been. Is that not working? Quite a lot of studies have been done to address the effects of offshore wind energy. But we dove a bit deeper, and we look mainly at the effects of the transport sea cables that are associated with these wind farms and the electromagnetic fields that are created around them. Elasmobranchs are very sensitive to both electric and magnetic stimuli. And if we look at the electromagnetic fields that are created around these subsea sea cables, you see that it's, there's a lot of variation in time and in the exposure, and it's multidimensional. It's a very complex total that they can perceive. So it creates an electric field. You have AC and DC cables, so that already gives variation. You have a magnetic field around it, and you have electric pulses coming from the cables themselves, but also induced electric currents by seawater ions running through the electromagnetic field. So they also vary with tidal currents, and also the amount of current power that's been transported varies with low and light wind conditions. So this is a very complex and variable exposure that elasmobranchs perceive, both from an electric and magnetic point of view. So the sharks and rays, they have a unique ability to detect minute electric currents. So the level of currents we're talking about here is way lower than, for instance, is used in deterrence. It's more on the level what they use ecologically than that. And yeah, we think that, to go back, we think that the electric pulse is mainly used in feeding ecology, connection to conspecifics or at close range. Whereas the magnetic fields is more used as a navigational orientation cue during longer migrations or large-scale movements. So we did a screening of the presence of elasmobranchs in offshore wind farms along the Dutch coast by using eDNA. And yeah, we found throughout the year presence of different species in and around these offshore wind farms. So they're using these farms as habitats. Surprisingly, we didn't find small spotted cat sharks, so it's still a bit puzzled why we didn't find them there, because they are in the area. And we did, but most of the work of Annemiek was controlled experiments to look at the effects of electromagnetic fields. One possible pathway how they are affected is by already in the egg case during the development. If egg cases are deposited close to sea cables, they are constantly exposed to electromagnetic fields. And already in 1/3 of their development, they develop these ampullae of Lorenzini, so they're already very early sensitive to also electric stimuli. And we looked at whether there was differences between in a controlled environment versus exposure to AC cables in these experiments. And we mimicked electric magnetic fields that are comparable to the ones you can find in a field situation. It was already known that when electric stimuli can cause, they ventilate with their tails to keep oxygen levels high enough in the egg case, and that when electric stimuli can cause freeze reactions to stop their ventilation. But we found in our experiments no indication for that. In fact, even the other way around. We found that in the exposed individuals, they were more sensitive in assessment trials and showed more hyperactive behavior under electric stimulation from these electromagnetic fields. So yeah, this might, perhaps it might also interfere with predation risk of that they're being more easily noticed. And also we saw that in the course of the trials, especially in the exposed group, we saw that there was divergence in individual responses to these electric fields. So that was also a puzzling result. We also looked at their development after hatching for a 3-month period and how they further developed. And we found no differences in survival and in growth from the exposed and unexposed egg cases, both in rays and in small-spotted cat shark. But we found some subtle morphological differences, so that the rays had a bit smaller tails and had a bit longer pectoral fins in sharks. So we don't. It's an intriguing result, but we don't know the underlying mechanisms of it and how it holds if we do more experiments on this. We also did experiments in larger tanks with adult skates and small spotted cat sharks. And there we found that In direct current, there was we didn't see strong responses that they avoided. The tanks were 15 meters long, and there was a gradient of an electromagnetic field generated in the tank from near field cable to hardly noticeable at the end of the tank. And we didn't find strong attraction or avoidance of these electromagnetic fields. We found more subtle differences. Okay, and it was we found lower activity in direct current and no change in behavior in alternating currents. We did find some subtle indications that there are, for a sex-dependent difference in behavioral response to within these electromagnetic fields, with lower activity in AC and DC for males and a bit higher activity for females in 50%, about 50%. Okay, I'll skip this one for. Oh, sorry, that's a bit too fast. We also did experiments in where we combined habitat preference and exposure to electromagnetic fields. So we had a circular tank with alternating mussel bed and bare bottom, where one half was exposed to electromagnetic fields and the other one was not. And here we found no preference, or they were quite indifferent to the electromagnetic field. So yeah, I can imagine that you're a bit overstimulated. That is how Annemiek also felt during her PhD. There's a lot of different aspects to look into. And the takeaway is that keep in mind that these electromagnetic fields around sea cables are complex, variable, and multidimensional. I would compare them to underwater noise exposure, which also has many features and different parameterizations. We didn't find any strong effects, either avoidance or and mainly subtle effects. So it's still pioneering work and a lot more to do, but I'll leave it to that. Then I'm happy to answer questions in the coffee. I think. Yeah.
Thank you very much.
Right.
Up next, we have Giorgio Fedele presenting Recon for a Dream, Circadian Patterns and Steep Light States in Side-Swimming Hammerhead Sharks.
Good morning, everyone. I'm gonna slightly change topic now. I'm gonna talk about sleep. Forgive me for the pun. So sleep is a highly conserved animal behavior. To date, there are no truly sleepless species. That highlights the importance of this sort of behavior, and it's critical yet not fully understood role in maintaining biological fitness. Yet, sleep is very heterogeneous. It can, in terms of its duration and architecture. To one extreme, for example, there are certain migratory birds and some marine mammals have evolved uni-hemispheric sleep, whereby they can sleep, whereby only half of the hemisphere sleep, of the brain sleep, while the other one remains awake. Despite all this variability, though, we can define behavioral sleep as by three key criteria: reduced activity of metabolic output, reversible diminished responsiveness to external stimuli, and homeostatic regulation, meaning that the more prior wakefulness or activity leads to a greater need for sleep afterwards. So the question is, how do sharks that are forced to continuously swimming sleep? Hammerhead sharks, scallop hammerhead sharks are actually providing an ideal natural test case, because besides being nocturnal obligatory run ventilator, they've been shown to use energy-saving swimming strategy called side swimming behavior, whereby they roll onto the side while keep swimming, and this posture is considered to is thought to reduce drag while they're moving, therefore reducing cost for transportation. But most importantly, this behavior occurs on repeatedly throughout day and night, providing a discretized behavioral signal in an otherwise continuous locomotion activity. So the question is, can we use this side-swimming behavior to test behavioral sleep? In order to test this, we tagged 6 adult males hammerheads off the coast of Hawaii for around 20 days across different years. And we basically segmented thousands of side-swimming behavioral bouts, and using unsupervised clustering algorithm based on the structure of those bouts, we found that actually there are 2 different states of side-swimming behavior. The first one, of course, predominantly during the night, and significantly diminishes during the day, and is characterized by longer bouts and an increase ODBA, or total activity, which is also our proxy for metabolic output. Whereas the other behavioral state, what we call low activity SSB, is predominantly diurnal, is characterized by by a shorter duration and lower ODBA, which is perfectly in line with the first hallmark of sleep, so reduced activity. However, reduced activity on its own is not enough to define sleep. So we next asked, can the activity of the night before, since those guys are nocturnal, predict what happens the following day? And it did. After more active nights, the sharks enter the low activity SSB significantly earlier the following day, and they stay in their state significantly longer, and also show a reduced ODBA the following day, which is consistent with the third hallmark of sleep, of behavioral sleep, which is homeostatic regulation. So to conclude, we think that this sort of side-swimming behavior is not just an effective locomotion strategy, but also it can underlie perhaps a resting or sleep-like behavior. The next steps would be to test the second hallmark of sleep, which is responsiveness or arousal threshold, and also investigating the physiological neurobiology underlying this sort of a behavior. In particular, we are interested in this, whether or not this lateralized behavior can be explained by asymmetric sleep, like in cetaceans and some migratory birds. Thank you.
Thank you very much, Giorgio. We do have time for some questions. Does anyone have any questions? We have one in the back. Thank you, Kristen.
Amazing talk. Thank you very much. Crazy study. Really, really cool.
Sorry, I can't see you.
Here, in the back.
Okay, okay. Yeah, sorry.
So the hammerheads can swim, like the side swimming behavior can go to either side. Is your hypothesis that based on the unilateral sleep, they swim on alternating sides?
Yeah, so that was my original idea. So one thing that I must say is that lateralized sleep is only being shown for mammals and birds. Reptiles, for instance, which are the next, well, the evolutionary step just below birds, they do not show lateralized sleep, and that's because they don't have REM and non-REM sleep. So the unihemispheric sleep only occurs during non-REM sleep, which is again peculiar of birds and mammals. So, but it could be still, yes, the contralateral hemisphere is probably not completely unihemispheric, but yes, it can be asymmetric. So yeah, the idea is that perhaps when they are tilting one side, that part of the brain is more active than the other.
Thank you very much, Giorgio. Sorry to cut you off. We have up next Joel Gayford speaking about shape-shifting sharks and ontogenic allometry in conjunctions.
Hey everyone, my name is Joel Gayford, and today I'm going to be talking to you about allometric growth and morphological scaling in sharks. So morphological scaling is all about the relationship between shape and size. And as animals grow, we can broadly define two categories of morphological scaling. First, we have isometry, or isometric growth, which describes simply the scenario in which shape does not change with size. This is illustrated here by the lizards at the top of this graph. And basically, in this case, adults are simply larger versions of juveniles and otherwise identical in terms of shape. Alternatively, we can see allometry or allometric growth, which describes substantial changes in shape with size. My favorite example of this is the human head, which shows pretty significant negative allometry. If you think of a newborn baby, they have really large heads relative to their body size. And as the baby grows, the head becomes smaller relative to body size until reaching roughly adult proportions. This is an example of negative allometry, but we can also see positive allometry as well. As far as sharks are concerned, recent studies have looked at patterns of morphological scaling in a number of species, finding that some grow relatively isometrically, whereas others, such as the bull shark, show quite profound changes in shape with size. The question then is why we see such differences in growth trajectories among shark species. The prevailing hypothesis is the allometric knee shift, or ANS hypothesis, which suggests that allometry is related to ontogenetic shifts in trophic or spatial niche. And the way this works is that in many species, adults and juveniles occupy quite different ecological niches, with adults generally experiencing lower predation pressure and exhibiting higher dispersal than juveniles. Where these shifts are significant, we would then expect adult morphology to be optimized for long-distance energy efficiency, whereas in juveniles, morphology might instead be optimized for maneuverability or agility. The ANS hypothesis thus predicts this bidirectional relationship between morphology and ecology, with the magnitude of shape changes we see over ontogeny being related to the magnitude of niche shifts. And importantly, whilst there is circumstantial evidence for this hypothesis, it hasn't been tested directly until now. So to test the ANS hypothesis, I traveled around the world going to museums and measured the fins and body of over 4,000 sharks comprising 67 species. For each measurement and each species, I then performed separate linear regression analyses to calculate deviation from isometry in each case. For each species, I then performed a comprehensive literature review to categorize them in terms of the presence or absence of trophic and spatial niche shifts, and finally compiled all of these data into Bayesian mixed models, which are a bit complicated, but just tests the base assumptions of this ANS hypothesis. The project's still ongoing, but I have some preliminary results to show. Here, this plot shows the effect sizes of all of the covariates included in my model. I don't have time to go through all of them, but the critical result is the bottom row, habitat L. And you'll notice that this covariate is negative and significant, which basically shows that the presence of habitat shifts are associated with substantial negative allometry in sharks, with providing good evidence for the ANS hypothesis. However, those of you who are more ecologically inclined might notice that this result is not only important for understanding differences in growth patterns among sharks. If there is a strong relationship between spatial ecology and morphology, then we may be able to use morphological data from fisheries or museum specimens as proxies to understand, predict, or inform aspects of spatial ecology of data-poor sharks or sharks that have been poorly studied. That's all from me. Thank you to all of these amazing organizations for helping fund and support my work.
Thank you very much, Joel. No time for questions. I'm sorry, you can catch Joel after. Up next, we have...
Okay, well, thank you very much. I'm here today to talk about this long-term project that we have in the Mexican Caribbean, trying to understand the biology and ecology of the bull sharks. So for this, I'm gonna talk to you about the Mexican Caribbean. We are part of the Mesoamerican Reef region, and in this part, we have around 85 elasmobranch species, and bull sharks are part of the fisheries there as target species, but also as a bycatch. And recently, like 10 years ago, there's pop up a very important tourism activity with bull sharks in the north part of the Mexican Caribbean that have led to some conflict between fisheries and tourism that move us to try to answer some questions and to try to give more information to improve conservation, trying to know more about the biology and ecology, and this is why this project started. So basically to try to gather more information, we use different approach. So we start monitoring the fisheries, but also doing some surveys between fishers and divers to know more how people interact there with the sharks and try to know about the distribution and habitat use of the species based on a citizen science program that we designed, and also based on the local ecological knowledge of the people there in the region. But adding more information, we wanted to know more about also the population genetics of these sharks there. We were very interested to know how the populations in the Western Central Atlantic were from this species. And recently, we include one of my students is developing a stable isotope study, trying to understand more the ecological role of the species there in the Mexican Caribbean. So our findings. Well, we have a lot of findings, but I don't have enough time to talk to you today here about all. But I just want to highlight two very important things that we found. First, our data, we were able to establish the first nursery area for the species in the Mesoamerican Reef region. So that it's the Chetumal Bay that is in the southern part of the Mexican Caribbean, and it's a very big bay, estuary bay, that is shared between Mexico and Belize. And what's most interesting is that with these sharks, particularly in this area, we found a very different population. So the baby sharks we were able to sample there were very different from the sharks in the north part of the Caribbean. So even they are very close, the population seems to be very different. And not only that, we recently, Nadia, that is going to talk tomorrow, we recently have discovered that actually is different from other populations in the Western Central Atlantic. So we have strong evidence of this new population in the Caribbean. And this leads to a proposal for an ISRA there that is moving forward. We have very good news that our reviewers are approving this proposal. So very soon we will have an ISRA there in this area. Thank you very much for all of you to hear this and for all my collaborators. And if you want to reach later, we can chat about more this project.
Thank you very much, Pilar.
Bye. All right. So, along the Kona coast of Hawaii, reef manta rays are predictably observed at 2 nighttime aggregation sites. And at both sites, divers place lights on the seafloor, which attracts zooplankton and in turn draws in feeding manta rays. But we wanted to know whether these nearby aggregation sites actually function the same way. So the north site, Gardenhill Cove, is a small sheltered cove, while the south site, Keaho, is a more exposed point along the reef edge. And these sites are located only about 20 kilometers apart and are both well-established nighttime dive sites. So by using 13 years of scuba-based citizen science identification data, totaling more than 23,000 sightings of 167 individual manta rays, we looked at aggregation structure, residency patterns, and prey relationships at the two sites. And despite being geographically close, they showed very clear differences. For example, the north site supported larger and more variable groups in approximately equal sex ratio. However, the south site consistently hosted smaller groups and was significantly female-dominated, with 91% of sightings being females. And this suggests that these nearby aggregation sites may be used differently. Residency patterns reinforce this difference. So at the north site, males and females showed similar patterns of use with regular return over time. Though at the south site, females were resighted much more consistently, while males were less frequently observed here, indicating stronger female site fidelity. And this helps explain the strong female bias in nightly sightings and suggests that the south site plays a more persistent role for a subset of females. These sites also differed in how manta abundance related to prey availability. So at the north site, manta numbers closely tracked zooplankton density, which would be consistent with the strong prey-driven feeding aggregation. And this relationship was weaker at the south site, suggesting that feeding may be only one of several reasons why mantas use that area. So to recap, the north site appears to function primarily as a shared feeding ground for males and females, while the south site may serve a more persistent role for females, where feeding is only one of several drivers of site use. And this may reflect cleaning opportunities, social behavior, or broader sex-based habitat segregation beyond nighttime feeding. And this shows that we cannot assume that nearby aggregation sites are interchangeable. So sites that appear similar may support very different ecological needs and face different vulnerabilities. So a one-size-fits-all approach is not sufficient, meaning that fine-scale site-specific research is critical, especially for island resident populations like Hawaii's reef manta race. And thank you to all my collaborators, data interns, and the Kona dive community for helping me make this possible. Any questions?
Thank you very much, Cory. Sadly, we have no time for questions. Next up, we have Aura Buenfil-Avila speaking about priority areas for conservation of large pelagic fishes in the Eastern Pacific Ocean.
Thanks for introducing me. Today, I am going to talk about one chapter of my thesis project. And so thanks for giving me this opportunity. In the Eastern Pacific Ocean, there is an important industrial tuna fishery that is represented mainly by port-sized vessels, and it's regulated by the Inter-American Tropical Tuna Commission. The Commission has implemented different conservation efforts, but the challenge remains in the limit of the effectiveness of the spatial fishery management, mainly in reduce the bycatch and the fishing pressure on the target species due to increase in juvenile catches. So, for this reason, in this study, we tried to identify priority areas for conservation of 14 species of large pelagics, such as, that's included 4 tunas, 5 billfish, and 5 sharks. The latter two groups of which are bycatch and have significant conservation status. The planning models that were developed by using zonation software, which took 4 factors into account. The first one is conservation target that is represented by the spatial distribution of the 14 species. The condition layers that is a reference of the optimal habitat condition for this species, and in this case, we use ocean fronts based on chlorophyll. A cost layer associated with the human activities. In this case, we used fishing efforts and pollution from maritime transportation. And a relative importance scores for each species based on its IUCN status conservations. It's important to mention that the planning models were applied under 3 environmental scenarios called neutral and warm. The mainly areas with the highest values of biodiversity and priority were associated with areas with low degradations and optimal habitat conditions, such as the western coast of Baja California Peninsula, upwelling zones of Central America, equatorial waters, that's included oceanic islands such as Galapagos, Malpelo, and Cocos. And also the results reveal that the spatial dynamics on priority depends on the environmental conditions, with the greatest change on the warm scenarios. An example for an area with high variability under the warm scenario is the upwelling zones on Central America. this area is considerable as a vulnerable in from the rising of sea temperatures, which can affect the availability of the habitat for these species. So these results reveal that the spatial management of these species need to be dynamic. I want to thank to all my collaborators that makes possible this project. And also, if you have any question, please contact me by email or in my research guide. There is the papers about my thesis project. So thank you very much.
Thank you very much, everyone, for coming.
Okay, good afternoon, everyone. Thank you. My name is Iqbal from Konservasi Indonesia. We would like to present our latest publication on whale shark ecology that we try to understand the niche partitioning. Whale shark often described as the fast animal, yeah, and they are moving in the fast dynamic ocean. However, we never know about the driver factor on pattern, the movement, and the habitat use. So one of our research questions is to understand what actually the driving factor in different life stage, sex, and also behavior. In Indonesia, whale shark frequently interact with the leaf net fishing. This unique interaction make us very easy to take the whale shark, as you can see in the video. By collaborating with the fishermen, we can take more than 70 individuals in the past decade. And this opportunity make us is very have a good resolution of data. More than a year of taking data we have, and some of them is until 3 years. And actually, we can retake the individual if the animal come to Bagan in the same time. Over a decade, yeah, we have 70 individuals we take. However, 71% they are juvenile male. Only 1 individual of adult male and 5 individual of female. However, the tracking duration is impressive. We can track more than 2 years of adult male and averaging 400 days for a female. The movement of whale shark in Indonesia is very impressive. They move across 13 countries and the high seas, representing one of the comprehensive data set for whale shark movement ecology globally. This is very critical because, as you know, whale shark in Indo-Pacific is present more than 60% of the global population. To understand why the driver of the movement ecology, we try to use the state space model, yeah, to differentiate the behavior state. And then we use that segregated data, including the region, different sex, demography. And then we use habitat model using seafloor geomorphology and oceanographic dynamic to understand the driving factor of the movement ecology in different region, different habitat and sex. And this is the result, yeah, the driving factor. For example, out of 160 model, we have 73 robust model that selected, yeah. The aggregation site are typically semi-enclosed water, productive water that very comfortable for juvenile male aggregate because there's a lot of food for them. However, in different contexts in non-aggregation area and very dynamic, they are complex seafloor geomorphology and become opportunistic foraging because they are characterized by front, ocean, thermal, and so on, Simon Canyon. And we observe clear ecological differences. Well, juvenile male influenced heavily by chlorophyll A concentration in coastal area, but slow flexibility strategies offshore depending on seasonal productivity. While adult male shift offshore such as the Southeast Indian Ocean, showing migratory behavior, potentially deep foraging near escarpment structure. The juvenile female prefer deeper offshore habitat influenced by sea surface height and canyon system. Finally, the ISO and MDBS show that the first one, the female is different structure with the male, yeah, where they are in the above, you see in the red cycle, while also the others behavior is different clustering with between foraging and migration. And also the bathymetric influence the structuring the clear niche partitioning. However, we didn't see evidence on life stage, chlorophyll A, and slope because maybe there's not become a clear pattern on niche partitioning. So in conclusion, what is matter for conservation? Because the MPA is very important for aggregation site. However, based on our data, this only protects small fraction of the whale shark ecological range. These species depend on connectivity that must integrate coastal aggregation site, deep sea migratory corridor, and seasonal foraging habitat. In short, we must move beyond static protection toward life cycle-based ocean management. Finally, I would like to thank to all funder and partner who have supported this project over a decade. And much of this research now is being published last week in the Frontier, so feel free to download the paper. And of course, I would very happy to discuss and looking for potential collaboration. Thank you.
Excellent timing, thank you. There's no time for questions, but on the dot 5 minutes. Okay, our next speaker in this session is Alicia Herrera. And I made a mistake in the beginning. It's actually 12 speed talks.
Good afternoon, everyone. I'm Alicia Herrera, and I will present a study frameworked in the project conservation strategies for shark and rabe in the Canary Islands. This study is a master thesis of Veronica Gonzalez, supervised by David Jimenez and me, and it's a study in the Canary Island, in Gran Canaria Island, in the southeast, studying the impact of microplastic in scallop megalops. Here, this is the region. We study the foul content of the stomach in 25 individual bycatches of scallop megalop. We digest the full content with hydroxide potassium, 10% for 48 hours at 60 degrees, and we found that 92% of individuals had microplastic in their stomach. Which kind of microplastic? We studied the morphology and colors, and we found that most of them are filament, lines, and fibers. 51% are filaments, 35 fragments, and 14% are films. A mean of almost 8 microplastic particles per individual. We also analyzed by micro-FTIR the polymer composition of these microplastics, and we found that most of them are semi-synthetic particles, and another part are synthetic particles. We studied by micro-FTIR because this is it's possible to determine a possible source of these microplastics. And in this study, most of them are cellulose, athlon, rayon, polyester, acrylic, ethylene. Most of these type of microplastic are in our clothes, okay? And the possible origin of this type of microplastic is wastewater discharge. In the Canary Islands, we have more than 200 points of illegal wastewater discharge. This is a possible source. Another type is the fishing gears, and also the oceanographic conditions in the Canary Islands bring up in our coast microplastic from the east coast of United States and Canada through the Canary Current. Our result is similar to other studies in other regions of the world. We know that microplastics are all over the world and all over the organism. And of course, our conclusion that this is an evidence that scallop megalop is a species highly exposed to microplastic pollution. 92% contain microplastic, but the type of polymer indicate the potential source of contamination. and we know, all of us know that this is not the main threat for sharks and rays, but we think that is a lack of information about the potential impact in the health, because it's not only the impact, the physical damage caused by microplastic, is the damage it's about the damage of different pollutant absorbent in microplastic, like additives and other that carried out in the ocean, like pesticide and other contaminants that transferred to the tissues, and really we didn't know the long-term impact in these species. And that is all. Thanks for your attention.
Thank you very much. I don't think there's time for a question there. We might run a bit behind. So we'll move on then. Our next speaker is Arielle Cowton.
All right, good afternoon. My name's Arielle, and I'm going to be talking about light deprivation in the epaulet shark. So who here is jet lagged? Maybe not anymore, but maybe on Sunday or Monday we were feeling the effects of light deprivation. We've come from many different time zones to be here, and changes in light exposure have a big impact on our daily activities. They influence our individual routines to coordinated behaviours across entire populations. So if light influences us, surely it also influences other organisms. Photoperiod regime is a fundamental dictating signal for living organisms to organize regulatory processes. And although embryonic development is often thought of as a genetically programmed sequence of events, in reality, its progression can be highly sensitive to environmental timing cues. Photoperiod regime has been seen to impact embryonic development rates, growth, and post-hatch lateralization in a number of species. However, its effect has remained largely unexplored for elasmobranchs, particularly those that develop in benthic egg cases, like the epaulet shark. Epaulet sharks are a small, benthic, reef-dwelling species known to exhibit exceptionally high tolerance to environmental extremes. However, our understanding of their embryological development or tolerance to various environmental conditions pre-hatching remains a mystery. Defined by Musa et al. In the greater spotted cat shark, and proposed as transferable across related species, including the epaulet shark, as seen here. Embryonic development in oviparous elasmobranchs has been described as following a 7-stage progression defined by key developmental milestones. Occurring during stage 4 of development, one of the most important milestones is the transition from tail oscillatory ventilation, where oxygen uptake is passive, to buccal pumping, where the embryo actively ventilates water across the gills and when space becomes a limiting factor. This key shift in respiratory strategy becoming the main point of investigation for this study. So, using the epaulet shark as our model species, we asked how does the absence of light cues alter the timing and coordination of key embryonic morphological, respiratory, and energetic milestones in oviparous elasmobranchs. Eggs collected from a resident breeding colony were incubated under 2 different light regimes, a baseline 12-hour light-dark cycle and a 24-hour complete darkness treatment. Eggs were examined non-invasively 3 times a week via candling, whereby we quantified developmental staging, respiratory strategy, and embryo growth and yolk consumption. Under baseline day-night photoperiod, embryos progressed through the 7 key developmental stages as expected. And in complete darkness, while there was no overall shift in developmental timing or stage duration, the onset of each stage was significantly accelerated. As you can see here, the transition from tail oscillations to buccal pumping over time, under baseline photoperiod, embryos' respiratory strategy occurred during stage 4 at approximately 10 weeks. However, in complete darkness, this transition was considerably earlier, with both the onset of buccal pumping and the transition to it as the dominant respiratory strategy occurring approximately 3 weeks earlier, representing a substantial shift in a key physiological milestone. Growth and yolk results also varied. However, in the interest of time, I'm not going to delve into them today, but if you're interested, come find me later and chat. These results demonstrate a substantial shift in a key physiological milestone, darkness acting as a signal that alters the internal developmental pacing of embryonic developmental elasmobranchs, highlighting a potentially important mechanism linking environmental light conditions to early life performance in reef-associated sharks. The timing of respiratory transition is directly linked to oxygen demand, metabolic rate, and readiness for post-hatch survival. Therefore, in environments where non-natural photoperiod occurs, whether it be increased light from artificial sources or habitat degradation or reduced light through sedimentation, the timing of key physiological transitions may either be accelerated or prolonged, impacting the future survival strategies and persistence of these species. Stage progression observations demonstrated that the development is retimed in a stage-specific manner under altered light conditions, reinforcing that photoperiod acts as a developmental timing cue rather than a uniform growth driver. This research demonstrates that photoperiod is not just a background environmental cue, but a timing cue that can reshape when critical physiological transitions occur and inform survival. Thank you.
Thank you, Arielle. Again, we have no time for questions. The next speaker is Abdi Hassan.
Hello, good afternoon all. My name is Abdi from Konservasi Indonesia. Today I will talk about our study in Indonesia, habitat segregation and residency of whales across the Sunda Banda and by fin-mounted satellite track. Understanding how threatened species move is essential for effective conservation. Animal movements are driven by the need to find food, sustainable habitat, and survive its changing environment. The whale shark, the largest fish in ocean, is now classified as endangered with significant population decline across the Indo-Pacific. Although this species is fully protected in Indonesia, its movement pattern and habitat use remain poorly understood. The study aims to fill that gap by using satellite tracking to examine how whale sharks move, where they spend time, and how they use the different habitats. Ultimately, this knowledge is critical to support better conservation and management strategies. Before looking at the result, let me briefly highlight the dataset. We deploy 68 satellite tags and 66 successfully transmitted data. After excluding one malfunction tag, we analyzed move data from 65 sharks. Most of the tag individuals were 61 male and only 4 female, with size ranging from about 3 to 9 meters, and tracking duration was quite extensive, ranging from just a few weeks up to more than 3 years, with an average around 1 year. Importantly, 38 individuals were tracked for more than 1 year and some for over 3 years, providing rare long-term movement data. Here are the results. Stats space model clearly show how whale sharks concentrate their activity in area with low movement persistence. They are likely activity foraging. These hotspots include major aggregation like Cendrawasih Bay, Kaimana, Saleh Bay, but also extend with wider regions such as Floresi, Banda Art, and Papua Coast. Additional feeding areas were found along the south of Java and even as far as Marshall Island and Papua Guinea. This tell us one important thing: whale shark feeding ground are widespread, and conservation effort must reflect this large spatial scale. Tracking data show clear seasonal pattern and show site fidelity in whale shark. They stay year-round in Cendrawasih Bay, while in Kaimana, sharks were present mainly from November to April, then move toward the Banda Arc between May and October. Importantly, our target sharks return to their original tagging site, showing repeated visit over time. 2 key hotspots stand out: Kotisori in Cendrawasih Bay and central area of Saleh Bay. Although each region has distinct home range, some overlap occurs in the Banda Sea and Flores Sea. Overall, this highlights both strong site attachment and wide-ranging movement patterns. Now, and the conclusion, In Indonesia water, especially between the Sunda and Sahul Shelves, are critically habitat for juvenile whale shark. They show strong residency in K area while also undertaking seasonal and long-distance movement, reflecting both site fidelity and ecological flexibility. As their range extends beyond protected area, effective conservation demand, expanded protection, open sea management, and international collaboration align with the scale of their movement. We gratefully acknowledge our donor, partner, and all contributors who support and collaborate made this study possible. Thank you.
Thank you very much. There's, again, no time for questions. Our next presenter is Eleanor Greenway.
Hello. Oh, let me stand a little bit closer. Hello and good afternoon, everyone. And I'm Eleanor Greenway, and I'm going to be presenting to you today about the daily movements of these three skate species in the Northeast Atlantic. QR code is on the top. I'm not going to be presenting about methods here, just the main results. So if you need more information, just click on the QR code above. For this research, I use mainly tagging data and stomach content data. And I know Steve is in the room, and yes, we tagged teenagers and adults. So we use data storage tags for our research, and this recorded mostly depth and temperature data. And I'm sure we've all seen like these plots over the past few days. So we have time on the bottom and depth along the side. And we see these skates really make a lot of use of the water column. And this is something we didn't quite expect. We traditionally thought these animals were really benthic and expected them to stay on the sea floor. But if we zoom in a little bit on the data, and hopefully you can see the colors on the bottom, we have orange representing daylight hours and blue representing nighttime hours. And we can really see they're on the seafloor during the day. And you can see this kind of wave on the data, and this is high tide and low tide. So then we know they're on the seafloor during this time. And they're almost exclusively swimming through the water column during these nighttime hours. So key take home here, seafloor during the day, swimming through the water column at night. And we know a lot of marine taxa also show this behavior. It's normally in relation to predator and predator avoidance behaviors, where animals are kind of close to the seafloor during the day where it's darker and a lot safer. And then during nighttime hours, they come up close to the sea surface where they can feed. And this cascades through the food web where predators are following their prey. So I wanted to ask, are skates also following their prey throughout the water column? So for this, we looked at stomach content data. So we only had the data on 2 species, so unfortunately we didn't have any data on the last species. And we looked mainly at stomach fullness. So are these skates feeding at different times of day? And yes, they are. So they're feeding during day, dawn, and dusk, and night. But they're mostly feeding, or feeding intensity is highest during dusk hours. And when we look into the literature, this is also when prey activity is highest. So they are feeding at different times of day, but mostly during these dusk hours. So, in conclusion, skates are on the seafloor during daylight hours and swimming through the water column at night. Feeding intensity is highest during dusk, but they are feeding throughout different times of the day. So this kind of shows us that skates are following their prey items throughout the water column during these daylight hours. So, I know it's very brief, but thank you for listening. If you need more information, scan the QR code above. If you wasn't in Hall B, then this is fairly new information to you, but I submitted my thesis and I'm defending on the 10th of July. If you want to join my defense online, there is more information on the link as well. It's also my birthday. We're having drinks at Fire Beach tonight from 8 o'clock, so feel free to join. Thank you.
Well done, Eleanor. And your timing is perfect to fit all that in with minutes to spare for one short question. One quick question. I will. No questions? Okay. Well then, happy birthday. Yeah.
Hi all. So for the first half of my PhD, I looked at movements of whale sharks at Ningaloo Reef in Western Australia, which, like many other aggregations, is dominated by juvenile males. And so the unique strength of the data set I looked at was that there was roughly even numbers of males and females across a wide range of sizes. 24 of the tags also reported vertical information for up to 325 days, and so this enabled us to explore their vertical movements throughout their migrations. So looking at the tracks, we can see there was some fairly variable movement strategies amongst individuals, but generally the animals tended to move north after they dispersed from Ningaloo Reef. They're likely following ephemeral patches of prey and remaining within their temperature range, so they then move south in the summer months. As you can see, there's not really any clear patterns between males and females, but looking at the vertical information, we can start to explore some more of their behavioural differences. As we can see, individuals spent most of their time in the top 10 metres of the water column, but they also regularly dive to depths beyond 1,000 metres. Again, the patterns look fairly similar between males and females, but there was a statistical difference, and so I investigated this further, particularly focusing on the top 10 metres, their surface use, using GAMs to explore the drivers of this behaviour. And so there was a seasonal pattern in males where they actually spent less time at the surface at Ningaloo and then used more surface when they went offshore. And this wasn't clear in females, but the most interesting pattern was the effect of SST on their surface use. And so in males, there was a clear reduction in surface use as SST increased, and this wasn't apparent in females. There was also a very interesting difference between sizes where larger females spent more time at the surface compared to smaller females, and this was actually the opposite in males, which is likely related to foraging as smaller males grow. So in conclusion, this is just one of the a general theme of sex-based differences among these individuals that were tagged at Ningaloo Reef across a wide range of sizes, including some mature individuals. And these patterns start to suggest some differing movement and or life history strategies between the sexes. This is likely related to varying growth rates and foraging requirements, and you can read more about it in the paper at the QR code. Thanks very much.
Stay there, you might get a question. Thank you. We have time for one question, maybe even two.
Thanks for the talk. So you do see some differences, but they seem relatively small compared to the other talk that preceded you, which saw, completely different patterns, although measuring very different variables. Can you try to maybe explain those differences? Why one talk would see one thing and yours another?
I suppose there's just such a wide range of movements in this particular data set, and the depth information was also aggregated daily, so it was hard to tease out sort of instantaneous drivers, and that may well be why the results are sort of smaller in variation. But the models also accounted for location, which does obviously explain a lot of variation, so the general trends are stronger in isolation. But yeah, thanks.
Are there any other questions? We have time for one very quick additional question. No. Okay. Thank you very much. Well done. The next presenter will be Sabrina Daly.
Good afternoon, everyone. My name is Sabrina Daly. I'm a fourth-year PhD candidate in the Block Lab at Stanford University, and today I'm going to be talking to you about our work on the intraspecific site fidelity and niche tracking of salmon sharks in the northeastern Pacific. For mobile animals like salmon sharks that live in dynamic ocean environments, they need to move to stay within their ecological niche and meet their energetic and reproductive needs. And to do this, they must select for habitat. And this requires making the trade-offs between the costs and benefits associated between selecting for familiar habitat that has known habitat resources and risks, and those associated with unfamiliar habitat which has unknown habitat resources and risks. When an individual animal chooses to return to familiar habitat, this behavior is known as site fidelity. Site fidelity is a common behavior across taxa, seen in both terrestrial and marine environments, with individuals showing fidelity to aggregation sites, regions, and what I'm particularly interested in is fidelity to migration routes. This is particularly pertinent as climate change poses the risk that previously familiar habitat with known resources may become unknown, meaning that site fidelity may become a less advantageous strategy under future climate regimes. But to understand that, we first need to know if site fidelity is advantageous under current climate conditions, and if it allows individuals to stay within a more narrow ecological niche. To answer these questions, we use data from 14 spot-tagged female salmon sharks, each recording data for over 2 years for each individual, allowing us to explore interannual variability of site fidelity behavior. We built movement-based kernel utilization distributions for each year of an animal's migration, and then quantified the amount of overlap between each year using Bhattacharya's affinity, with an average BA of less than 0.6 being associated with low site fidelity behavior, as seen here in the figure on the left, with low overlap between each year of the migration, and an average BA of over 0.6 being associated with high site fidelity behavior, as seen here in the figure on the right, where there's high levels of overlap between each year of an individual animal's migration. What we found is that there's clear evidence of intraspecific variability in site fidelity behavior within this population of tagged salmon sharks, with 10 individuals exhibiting low site fidelity behavior and 4 individuals exhibiting high site fidelity behavior. We also extracted environmental covariates along the tracks of these animals to look at the environmental conditions experienced by different individuals. We found that low site fidelity individuals targeted warmer, saltier, more turbulent water, likely indicating they're exhibiting more searching behavior and taking advantage of opportunistic foraging patches, while high site fidelity individuals targeted slightly fresher, more stable water masses, likely returning to the same stable environmental features like seamounts and river mouths, where they can find reliable prey sources. In regards to the ecological niches of these animals, we found that low site fidelity individuals have a wider niche breadth compared to their high site fidelity counterparts, with high site fidelity behavior allowing individuals to track a more narrow ecological niche. Here we see a wider niche breadth by individuals that exhibit low site fidelity behavior across seasons in relation to their thermal niche, looking specifically at sea surface temperature. And we saw similar patterns along other environmental covariates like salinity and eddy kinetic energy. So in conclusion, the key takeaways of this work are that there's intraspecific variability in site fidelity behavior in salmon sharks, low and high site fidelity individuals are likely targeting different environments, and high site fidelity behavior is allowing individuals to track a more narrow ecological niche, with the next steps for this work planning to look at conditions in areas of high move or low move persistence to determine if conditions at potential foraging sites differ between individuals that exhibit these various behavioral strategies. And with that, I'd like to thank my co-authors, collaborators, and funders, and I'm happy to take any questions.
All right, thank you very much. There's time for one quick question. Are there any questions? No question. Oh, there's one at the back there. In the meantime, can I ask if Aura Buenfil-Avia is here?
Hi, Sabrina. Thank you very much for your presentation. It's really nice. I wanted to ask you, have you seen any other variable between the ones that have site fidelity or not? Like, did you see if any had more fitness than the other ones, or any other difference that they're being more successful for having niche fidelity?
Yeah, thanks for the question. So we looked at movement, specifically looking if there are variabilities in the movement patterns of different individuals and across the different behavioral strategies, and we didn't really see much differences within the wider movement patterns of the salmon sharks. The reason we're looking at this specifically with salmon sharks is because there's so much variability in the migration routes within this population that we're able to look specifically at the behavioral strategy. But in terms of fitness behavior, we didn't really have that with the spot tag data. So we just looked at it in regards to the movement.
Are there any other questions? We have a little bit of time still. No other questions. Okay, thank you very much. Our next presenter is Jessica Schulte.
Hello. Hi, everyone.
My name is Jess Schulte, and I am a fifth-year PhD candidate in the Chapel Big Fish Lab based at Oregon State University. And today I'm going to be giving you a very high-level look at my PhD research that I've been conducting on broad-nosed seven-gill movement and foraging ecology. So, seven-gills are large apex predators that live in temperate coastal ecosystems around the world. They can be found in ranges within the continental shelf in waters typically less than 200 meters. And the adults and subadults will exhibit this really high seasonal site fidelity in certain bays. They are generalist and opportunistic feeders, and they are considered apex predators and very important species in the ecosystems that they're found in. But unfortunately, very little is known about them in the Northeast Pacific. So, take you over to my study sites, Willapa Bay and Puget Sound in northwestern United States in Washington State, which are two large and important estuaries in the region. They're home to important commercial fisheries, as well as managed species such as harbor seals and other marine mammals, Dungeness crabs, salmon, and conveniently for me, high seasonal abundance of seven gills in the summertime. So what I've been doing is heading out during the summer during their annual seasonal abundance between the months of March and October, and I'm catching them, bringing them on the boat, and then I'm collecting morphometrics, a bunch of tissue samples, I'm collecting stomach contents, and then I'm also tagging them externally with conventional tags. All of them get the conventional tags, and then a subset of them will get internally tagged acoustic tags. So far, or I guess over the last 4 years, I've sampled over 330 individual seven gills, and so these are all adults or subadults, ranging in size from 98 centimeters all the way up to just under 3 meters, and about an even mix of males and females. And of those 338 individuals, I've just had 12 recaptures, so very low recapture rate. Looking at some of the acoustic results, above the dotted line are all of the individuals that have been tagged in Puget Sound, and then below the dotted line are all of the individuals tagged in Willapa Bay. The blue are the individuals detected on the Puget Sound array, and then the green are detected on the Willapa Bay receiver array. And you'll notice that there's very little mixing between the two. So individuals that are tagged in Puget Sound are not traveling down to Willapa Bay and vice versa. Yeah, there's that difference between them. But you will notice that they are arriving at the bays at the same time each year, separately. And it turns out that that timing aligns with seasonal abundance in prey, so that's what we're starting to think, that it's food-related. What else did I want to say? Oh, and the last thing I wanted to mention is that even though sharks are largely disappearing once they leave out of Willapa Bay, or out of either of the bays, we did have one individual that was caught by a fisherman down in Southern California, and then another one that was tagged with a pop-off satellite tag showed up in Southern Alaska. So even though they're largely disappearing from this array, these arrays, it seems like they are exhibiting these high seasonal migrations and then returning back in the summertime. There we go. So next thing I want to talk about is my stomach contents. So to provide an immediate snapshot of diet, I've been collecting stomach contents using a lavage technique on the boat. So I hope you guys are hungry, because here comes some shark puke pics. So these are some of the interesting samples that I've collected from the seven gills. On the left was an extremely large sample of harbor seal collected from a very small individual. I've also collected what I thought was going to be sea otter, but turned out to be beaver. And then down here, I thought it was going to be river otter, but it ended up to being American mink. So they seem to be eating a little bit more terrestrially than what I thought they would be eating. Looking a little bit more at the stomach contents in general, I've been identifying the stomach contents visually and weighing them. And so as you can see, the vast majority of what I've been collecting in my stomach contents have been marine mammal, and specifically harbor seal. And so, like I said, this is by weight, but I'm also seeing similar trends by abundance as well. So the vast majority of what they're eating is marine mammal and mostly harbor seal. So what does this all mean? There hasn't really been a lot of studies on seven gills in the Pacific Northwest, and there's very little known about them, but it seems that they might be exhibiting a much higher role in the ecosystem than has previously been thought. So keep an eye out for my results. I'm in my 5th year of my PhD, so I'll be defending soon, and you guys are all invited to my defense. Thank you. Thank you.
Thank you very much. Okay, I think there's no time for questions. The next speaker is Stephen Kajira.
So every winter in South Florida, we have thousands of black-tipped sharks that come down and spend the winter in our warm waters just off the beach. This gives you an example of what it looks like. Every one of those dots is a shark about the same size as me, about 174 centimeters long. And they are literally right up against the beach. You could be in your backyard and look out and see the sharks. As marine biologists, we can't afford houses on the water, so we can't do that, but other people can. These are all black-tipped sharks, Cartilaginous limbatus, and like I said, they come down every winter, and what we want to do was enumerate how many sharks were here, when they were here, what was going on. And so we figured the best way to do this would be to do an aerial survey, and so what we would do is we'd fly along the beach and just cover, we're just going to focus on Palm Beach County, which is Boca Raton up to Jupiter Inlet. We did all the way from Miami all the way up, but there's hardly any sharks down there, so we're just going to focus where the action is. We had a high-definition video camera mounted out the window of the plane, and what this enabled us to do is film the whole transect from Boca to Jupiter, from about the shoreline to about 200 meters off the beach, and count the numbers of sharks. So when you look at this, what you see is starting from 2011 to 2021, it's a 10-year span, you would get huge numbers of sharks in the wintertime, followed by hardly any in the summer. And then you get a huge spike again, followed by hardly any. This would continue year after year after year after year. And what I want to do is focus on what happens when you overlay water temperature on this. What I want you to notice is that you get the big numbers of sharks when the temperature is cooler. Then the water temperature is warmer. What happens? Lower numbers of sharks. You got this inverse relationship between shark number and water temperature. Okay? And you can see this by looking at when we see the sharks. You only see the big numbers of sharks when the temperature is cooler, less than about 26 degrees or so. Okay? So, with this in mind, what happened over the course of the 10-year study was we found the average winter water temperature increased by about 1.2 degrees Celsius in just 10 years. That's a huge difference in a short period of time. All right? And so if temperature goes up, what do shark numbers do? They go down, right? And we saw that. We saw that the peak numbers of sharks went from over 12,000 to 1,200. It was decimated. The numbers went way, way, way down, all right? And if you look at what's happening, when we started this survey, you can sort of break the Palm Beach County apart by the different inlets. From Boca to Boynton Inlet, we'll show here in pink. Boynton to Palm Beach is blue, and then Palm Beach to Jupiter is green. When we first started this work, we saw most of our animals in the blue region. You always had a few from Boca to Boynton. About 3 to 7% of the sharks were down south, all right? But most of the sharks, when we first started, were in the blue area, Boynton to Palm Beach. By the end of that 10-year span, it had shifted, and most of the sharks were in the Palm Beach to Jupiter Inlet. They were basically shifting their distribution farther north. Basically, think of it this way. If you're migrating south, why bother swimming all the way down if your preferred temperatures are only part way down? You can stop there, be comfortable, no need to go all the way down. And that's what's happening. We're seeing this distributional shift in the sharks in just the span of a decade. I think it's a big change, a big temperature change, as well as a distributional change in a short period of time. And it really portends terrible things happening in the future. So with that, I'm going to wrap it up and acknowledge the Colgan Foundation. They funded our work for a long time. And if you're on social, by all means, follow us. Thank you so much. I'm done.
Thank you, Stephen. That was a great talk. Are there any questions for Stephen? Come on, someone must have a question on that stuff. Here we go.
That was a great talk. I'm worried about causation versus correlation here. If you plotted, you plot the curve of daylight hours, or lots of things change seasonally like that. How do you know it's temperature?
Yeah, it's a good point. We looked at temperature. We looked at productivity.
a bunch of different factors that came out. Underlying all of them, and prey abundance, underlying all of them is temperature. Temperature drives it, and it's the strongest correlate. You're right. It's not causation, but a strong correlation. And of all the variables that we looked at, this was the one that stood out the most.
Okay, thanks. We don't have any time for any more questions. Okay, our next presenter is Erwin Hendrik Winter.
Hello, everybody. I would like to talk to you about a monitoring program study we're doing in the Dutch Caribbean on reef-dwelling sharks, together with my colleague Guido Leurs. The study area is in the northeast of the Caribbean, the Dutch island St. Martin, Saberbank, and the Saberbank and Sint Eustatius. And the Saberbank is really one of the biggest atolls in the world. It's a unique system. We built an array of acoustic receivers in that area to study the behavior of reef-dwelling sharks. And you see that the three islands are well covered with receivers, but the Saberbank we could only, because of its huge size, we chose to put our arrays on the steeper slopes of the east side, but I would love to have a better coverage of the Saberbank. We started with the first pilot in 2014, and since 2015, we have a network in place of, on average, about 34 stations. And the stars notate times when we got out and also tagged sharks. Our main study species are four reef-dwelling species: Caribbean reef shark, nurse shark, silky shark, tiger shark, and in total we have now 120 sharks in the study. It's only 5 minutes, so I'm going to just give you a few bits and pieces, but we have, of course, way more than what I show you here. But what I found a remarkable result was for adult Caribbean reef shark. They had very small home range, only a few kilometers, like this individual was 924 days only seen on the south side of St. Eustatius in only a few kilometers. But once it had a very directed excursion to the Saberbanks. In 24 hours it came there, stayed for 7 days on the Saberbank, was only detected locally there. And then in 15 hours went straight back to his own home range and stayed there for the rest that we saw it. And this is a pattern we saw in more Caribbean reef sharks, both females and males. We speculated it's reproductive related, but of course, we cannot question them, what their purpose was for their voyage. Another example is, for instance, part of the Saberbank, Poison Bank. We attacked juvenile Caribbean reef sharks and also silky sharks. They live together in the same area. But they showed quite some habitat segregation. We saw that if you look at a daily pattern in how much detections we got, and we saw that silky shark were more detected during the day, and then less at night, whereas the Caribbean reef sharks were seen throughout the day. The number of detections did not change in the course of the day. And the silky shark used shallower water depths than the Caribbean reef shark, suggesting also a bit more pelagic water column use than the reef sharks. And what is also nice about acoustic telemetry is that you don't see your own animals, but you also get some nice surprise tagged animals from other studies. Like for instance from the US Virgin Islands, we saw 2 tigers and 1 nurse shark moving to our study area, over 150 to 200 kilometers over deep water. So for instance, the nurse shark is mostly considered more resident individuals, although in Florida they also saw some quite long distance migrations. But well, just want to show you that the study on St. Croix was from Grace Castleberry. So with it, I would like to stop just a few minutes. If you want to know more about those studies, come and see us, and I'll be happy to tell you more about it. I would like to thank all the cooperators and the local nature foundations that we work together with in a close cooperation. Okay, thanks.
Thank you very much. Time for one quick question. Are there any questions there? No questions. All right. Thank you very much.
Okay, Sean Liddy.
Thanks very much. Hi, everyone. So next door, there is a panel discussion about the very policy that might impact the trajectory of shark and ray populations. It is a big conversation with big impact, and by comparison, I might expect many of you to find my talk, which is about very small organisms on very small sharks, to be a bit less interesting. But you are all here, hopefully not only because your talk is on after mine, but because you are people that understand that it is from little things that big things grow. This research was conducted on Indigenous land and sea country, and I'd like to acknowledge the traditional owners, the knowledge holders, ancestors, and families whose connection to country and stewardship really sets a standard for sustainability that I think we can all learn from. So, why should we study microbiomes? Well, complex organisms, you and me included, we have outsourced most of our very important physiological functions to microbes. Our ability to extract nutrients from our food, our skin's immunocapacity, it all relies on things like bacteria. And we know that it is the first inoculation or the pioneer microbiome that has really enduring impacts on long-term health and function. And so this is pretty important when you consider our globally declining sharks and especially their very early life stages. But it begs the question, where do organisms get their microbiome? We have three main modes of transmission. So vertical, parent to offspring. We know this is really important for humans. Horizontal, think of the 3 F's of trophic ecology. You have foraging, feeding, and mating. And of course, environmental acquisition as well. So this is transference from habitat-forming species like kelp, also water quality, runoff, these kinds of factors. And we know that in sharks, they change the way that they interact with their environment, so their habitat use and their diet, a lot as they age. So that got me curious about the following question, which hasn't really been addressed in sharks. How do they acquire their microbiota, and how does this change as they age? To do this, I looked at the Port Jackson shark, which is an awesome species of benthic shark that aggregates in shallow coastal waters in Australia, in temperate Austral and spring during mating aggregations, and they show really strong site fidelity to these nursery grounds. Females lay a couple of eggs every few weeks in this season, and those eggs take usually just under a year to hatch. When they're first laid, they're closed to seawater, and it's not for the first couple months that mucus plug that keeps them sealed dissolves, and they get that first contact with the outside seawater. So this makes them a pretty perfect system to investigate this question, to see whether there is vertically transmitted microbiota from the parents or if it might come from their environment. So that led to my two hypotheses. That the albumin or the jelly of the freshly laid eggs will identify if there's any vertically transmitted microbes, and number two, that their microbial community structure, their microbiome, will change as they age due to variation in their ecology with ontogeny. So So to answer the first hypothesis, I collected freshly laid eggs from a field site and sampled the jelly with a syringe, and I did one of three things with that jelly. I looked at microscopy, I tried to culture any bacteria there on 2 different kinds of media, and I also extracted DNA from the jelly and then checked for any amplification. To answer my second hypothesis, we sampled the 3 different life stages of the Port Jackson shark, so those eggs that were open to seawater, where there were embryonic development already, neonates and adults, all from the same site, and I used a swab of the epidermis. In a previous chapter of my PhD, I found that this body region was no difference between sexes, so it was the most consistent across the life stages. I extracted DNA from these swabs and then did 16S rRNA amplicon sequencing before downstream analysis. Because of the biology of this species of shark, I was not able to sample all of the life stages simultaneously, but I replicated across sampling years to try and increase the robustness of the analysis. So what did we find? For the first hypothesis about the albumin, in the culturing, there was no growth on either media. In the microscopy, there was no microbial cells that could be detected. And in PCR and gels, there was no amplification. And I verified this with positive and negative controls. So altogether, this leads to the conclusion that the albumin of Port Jackson shark eggs did not include or did not contain any viable microbiota. Looking at the second hypothesis, so how does the microbiome change across life stages? This, to start, we have a few univariate metrics to look at. So we have bacterial richness and Shannon's diversity, and both tell the same story. So you can see in blue we have the adult richness and diversity, which is significantly lower than the embryo and neonate life stages. But again, this is very complex multivariate data, so we have to look at it in that way. And this is the NMDS plot showing variation in microbial community structure between these life stages. And it adds a really interesting piece to this puzzle. So we can see that despite there being low richness in the adult samples, there's a lot of individual variation. You can see that with the dispersion of these blue dots, each of which represents one epidermal sample from each unique shark. You can see that the high diversity populations were actually all really similar to one another, and this trend was consistent across both sampling years. So this is showing that data across sampling years. All pairwise comparisons were significantly different from one another. There's a lot more to the story than I can get across in just 8 minutes. So I could talk about the number of ASVs that are conserved across the development and which of those are unique to each life stage. I can talk about which ASVs actually, and ASVs are kind of like bacterial species, construct the pioneer microbiome, and how does the core, so those that are present across more than 80% of the samples, change across life stage. And I could also look at which ASVs drive significant differences between these life stages. So if anyone's interested, please come and find me afterwards. What are the key findings? So, for the first hypothesis, it's really difficult to prove the absence of something. But the lack of visible and culturable cells, the lack of PCR amplification as well, strongly suggests that the albumin could be sterile. So referring back to that question, it looks like there's no vertical transmission in Port Jackson sharks. And this means that their microbiome is likely to be environmentally acquired. For our second hypothesis, we can say that the microbial community structure does change across life stages, so with ontogeny. And the neonates are kind of the compositional bridge between the embryos and the adult life stages. We also saw that the adults are low diversity but high dispersion, so that means that there's a lot of individual variation. And what I think this means is that there's an environmental source population in the embryo life stage, and then that is subject to selective pressures and individual variation in diet and habitat use across ontogeny as that shark ages. Why does this matter? Well, Port Jackson sharks are not only very charismatic mesopredators, but they also show really strong site fidelity to nursery sites. Many of these in New South Wales are very highly urbanised, and they're warming faster than global averages. When you consider the importance of that pioneer early microbiome in the long-term health and function of sharks, this shows that we don't just need to manage fisheries, we also need to protect the key habitats that they rely on. These findings are also applicable to shark husbandry and breeding programs, which I think would be awesome avenues for future research. And to wrap it up, I'd like to thank all of the people that have supported me throughout my PhD, and also let everyone know that I'm now headed up north, and I work for Torres Strait Regional Authority, so if anyone is interested in working in this area, please reach out. Thank you very much.
Okay, thank you, Sean. We have time for literally one very, very quick question. Nope. Okay. So next up is Le Insabi.
Okay, can you hear me well? All right. Hi everyone, I'm Laurit Sabi, PhD student at Nord University in Norway. We're all here because shark conservation matters, and my talk today will take you from chromosomes to populations using genomics to better understand the management and conservation of the spiny dogfish. The spiny dogfish, once one of the most abundant sharks in the North Atlantic. In Europe, it became the fish in fish and chips, valued for its liver oil, and sold under all sorts of misleading names like rock salmon in England and salmonette in France. By 2005, the North Atlantic stock had collapsed to just 90% of its 1905 levels, and in response, fisheries were closed across much of the region. But today, even 10 years later, in 2023, the European stocks were considered recovered enough for limiting fishing to resume. But this recovery remains deeply uncertain, and the Mediterranean population is still listed as endangered. So all of this raises a question we haven't answered honestly enough yet. Do we actually know what we are trying to protect? Because until 2010, the spiny dogfish was considered one species worldwide. Then Ebert and colleagues looked more carefully and found that the entire North Pacific population, here in blue, was actually a different species, and they formally resurrected Squalus suclei. That same year, Verissimo and colleagues looked at, and they confirmed that deep genetic divergence happened between the Pacific populations and the Atlantic ones. And as you can see on this slide, these two sharks live very different lives. Squalus acanthias, in orange on this slide, matures at 5 to 8 years and lives up to 20, while Suclei doesn't reach maturity before 20 years and has been aged to over 80. In the North Atlantic, Acanthias is recovering slowly under strict management strategies, while Suclei is declining, even without being directly targeted. Last year, Davidson and colleagues documented a 51% coast-wide decline in biomass in the North Atlantic of the US, with the females being targeted. So at this moment, we have two species that were treated as one for half a century, now facing apparently different futures. But how are the species and populations connected across ocean basins, and how are they responding to their environment? To answer these questions, we need to look directly into the genome across many individuals and many locations. But to do so, you first need a very good reference genome. Few years ago, in 2023, my former colleague, Dr. Wagner, produced the first genome assembly for Squalus acanthias. It was a great start, but it had gaps and inaccuracies that needed to resolve. So we spent time doing that. And so what you can see on this slide is called a high-C contact map. And to make it simple, each bright block along the diagonal is a chromosome, and the better the assembly, the cleaner that signal. Next to it, on the left, you can see the genome for squamous nuclei that has been produced last year by the Vertebrate Genomes Project. So with these two reference genomes in hand, we sequenced 87 Acanthias, spanning almost the full range of the species, and we have 30 nuclei along the North Pacific of the US coast. All samples were sequenced using low-coverage whole genome sequencing, which is short Illumina reads at 5 times coverage, and analyzed with a pipeline specifically designed for those data. So first, let's look at what we see with both species. So this is a principal component analysis where individuals that are genetically closer appear together on the plot. Circles are Acanthias, triangles are nuclei. And as you can see, the first axis alone completely separates the two species. And so this is a genome-wide confirmation of what Ebert and Verissimo has been addressing in the past. So those are not populations of the same species. Those are actually different lineages. So at this scale, what we can see is that they are completely separated. And on the right, you can see that the southern hemisphere cluster together at the bottom. And on the top, we have the southern hemisphere of squamous Acanthias. So if we look closer on this, we can see that on the left, Argentina and New Zealand form a distinct cluster. And on the right, we have the Northern Atlantic with Norway, UK, Ireland, and the east coast of the US that are broadly connected across the Atlantic. On the PC2, otherwise, we have the Adriatic Sea that is apart from the rest, and admixture comes from this. At K2, we have a clean northern versus southern hemisphere, and the Adriatic is already showing a distinct signal, and at K3, it emerges as a distinct signal. So for nuclei, we see no structure, basically. Every points are scattered on the plot, and so we'll have to investigate this further. Heterozygosity tells us how genetically diverse each individual is across their genome, and two things stand out here. First, Acanthias show overall the higher diversity than nuclei, and given nuclei's biomass decline and that lower diversity is a finding we take very seriously from a conservation standpoint, and we'll have to investigate that further. The second position, then, within the Acanthias, the southern hemisphere population show higher diversity than the North Atlantic, which raises questions about effective population sizes and exploitation history. And so we'll have to look at that in our paper. So where does this leave us? These are two genuinely distinct species, genomically, ecologically, and in terms of conservation urgency. For Acanthias, the data show a clear north-south genomic split. Argentina and New Zealand emerge as distinct population units across the basin, which is, and the North Atlantic is broadly connected across the basin, which is consistent with the current ICES single stock management framework. And but we need to be cautious about that because actually the Mediterranean is still listed as endangered. And new samples from the Falklands are incoming, and this will help us characterize better the southern hemisphere population. And for nuclei, we cannot draw firm confirmation yet, as no population structure was detected across the North Pacific of the US coast. We have new samples from Japan that are incoming, and this will be key for understanding whether distinct management units exist across the North Pacific. So these genomic results are congruent with what Verissimo has been observing with microsatellites and mitochondrial DNA. But now the genome-wide confirmation allows us to go further. So the tools are there, the data is building, and there is real scope for genomics to inform the next generation of conservation decisions for these two sharks. So this PhD project spans 10 countries and only exists because of the people and the institutions you can see on this slide. So I would like to thank each and every one of you for your samples, your support, and your expertise. There's a few people that contributed that are in this room. You know who you are. So I would also like to thank the Darwin Tree of Life Wellcome Sanger Institute and the Vertebrate Genome Project. Thank you very much. It has been an honor to present this work to you today.
Okay, one question for Le.
A very simple question about the species identification. So based on external morphology, can you tell the two different species?
So there is a bit of difference in morphology. We have also some patterns and the vertebral counts that are different in those two species. I'm not an expert in Squalosoclei identification, but Fabienne Ziadi, which is in this room, is. And so I think we can contact her. Thank you.
Okay, thank you. We'll move on now to Jessica Pierce.
All right. Need me? Hi everyone, I'm Jess. I'm a PhD student at the University of Western Australia, and today I'll be telling you a little bit about the first chapter of my PhD, sequencing the first ever reference genome for a tiger shark and how we can use these genomic resources to investigate evolutionary and conservation relevant questions. And I'd first like to acknowledge that UWA is situated on Wajak Noongar Budja, and that this study was conducted using samples collected from Bayungu, Yeniguria, and Westland GC country. I'd also just like to quickly acknowledge that this work was funded by the Minderoo Foundation. I'd like to acknowledge the Oceanomics team for all their help, along with my supervisors and other co-authors on this project, as well as the Holsworth Wildlife Research Endowment for allowing me to attend this conference. So I don't need to explain to anyone here why sharks are important, but in case you're not familiar with this particular species, tiger sharks are distributed circumglobally around the tropics, and back home in Western Australia, in the UNESCO World Heritage Area of Shark Bay, they are a keystone species, performing critical top-down control via predation, as you can see in this video. They are often caught recreationally and are actively managed by our fisheries department, and they are also classified as near threatened by the IUCN due to a 30% global decline. But they are also a very interesting species from an evolutionary perspective, as they represent the last of their kind, with all other family members extinct. However, so far there has been no full genome sequenced for this species to my knowledge. So I don't have a lot of time to get into the technical details here, but if you want to know more, please come talk to me. This work was done through the Ocean Genomes Initiative, so also please find Shannon Corrigan at her poster later if you want to know more about that. But basically, to start with, I collected a snap frozen blood sample from a male tiger shark specimen from Exmouth Gulf in Western Australia, from which we conducted PacBio hi-fi long-read sequencing, Illumina short-read high-C sequencing, as well as Oxford Nanopore Technology ultra-long-read sequencing. With this combined data set, I ran it through the Ocean Genomes assembly pipeline with manual curation to assemble chromosomes, which resulted in a phased haplotype genome assembly. So for those who are interested in the assembly itself, here is a quick overview. We were able to assemble 86 chromosomes, so 42 autosomes plus 2 sex chromosomes aligning with the known karyotype. And I've been able to do a preliminary annotation of the genes, GC content, and repeat content, which you can see here. Now, as PacBio hi-fi and ONT data both have the ability to call methylation, I've also characterized the methylome for this genome, which to my knowledge is the first time this has been done for an elasmobranch species. All right, so just a few little specifics here. We were able to identify the X and the Y sex chromosomes, which is the first time anyone has shown that this species displays the sex system, as previous cytogenetic karyotype studies were unable to identify heteromorphic sex chromosomes at all in the species. So to do this, we resequenced a few known sex individuals at 50x coverage and compared the ratio of coverage between the males and females. So you can see all the autosomes, there's 42 autosomes, form a roughly 1-to-1 ratio between the males and females, while the X chromosome had double the coverage in females, and of course the Y chromosome was only present in the male samples. So the global methylation was an average of 63% across the whole genome, with gene regions being highly methylated and promoter regions being lower methylated, all of which is fairly characteristic for a vertebrate species. So it's nice to know elasmobranchs also follow the same pattern. And finally, phylogenomic placement matched our expected taxonomy compared to other known reference genomes for the Carchariniform order. So to start investigating the demographic history of this species, I first looked at inbreeding through runs of homozygosity. So this was under the principle that longer runs of homozygosity indicate more recent inbreeding, while shorter runs of homozygosity indicate historical inbreeding due to recombination events. So for context, I've added the great hammerhead shark, which has known high levels of inbreeding, and the short-fin mako, which had a historical bottleneck but has since recovered. And as you can see, the tiger shark follows a similar pattern to the short-fin mako, although a little bit lower, suggesting some degree of both ancient and recent inbreeding. So finally, I ran a PSMC model to reconstruct effective population size, or NE. And you can see that we initially have a very high NE around 6 million years ago, although tiger shark, the modern tiger shark species did evolve around 13 million years ago, so we are missing a slightly early portion of the picture here. So this was followed by a slow decline with the Pliocene cooling and the onset of the ice ages. Around this time, all other tiger shark species died out. However, our modern tiger shark species populations did kind of stabilize through the rest of the ice ages. At this point, we would have expected to see a population expansion with the beginning of the mid-Holocene warm period, but instead what we actually see is this almost series of bottlenecks and extended low population size, effective population size instead. So this was around 11,000 years ago that this happened. This was a period of geologically rapid climate shifts at the end of the ice ages with the Younger Dryas event, likely causing a mass change in ocean currents and food availability with this influx of fresh water. And while these results could suggest some reduced capacity for future adaptation, this low genetic diversity does seem to be quite characteristic for a lot of shark species, so it may not necessarily have any deleterious impacts. But the PSMC did show that tiger shark populations were sensitive to that climate shift, and this could potentially still be impacted by future ones. So inbreeding resulting in the shorter runs of homozygosity that we saw in the tiger shark likely resulted around a few thousand years ago, so this matches that time frame again. The longer runs of homozygosity may indicate some degree, small level of inbreeding in the past 200 years, but as there is no current effective population size for the Indian Pacific Ocean population, this is uncertain if they were able to rebound after this or not. This is why I'm now in the process of population-wide whole genome resequencing for tiger sharks around Western Australia and the Indian Ocean. So this is some very preliminary kinship results that I've been able to come up with. And as you can see, this is roughly 140 samples that I've been able to sequence, and roughly half of these samples were at least first cousins, even across samples between South Africa and WA. We even had 3 pairs of first-degree relatives, so potentially full siblings within this WA data set. And then for the last chapter of my PhD, I've developed a microhaplotype SNP panel for use with tissue and eDNA samples. So we're hoping to look at individual identification, contributing individuals, and assessing population genetics with non-invasive methods. All right, thank you for listening. Keep a lookout for these as this comes along. I'm writing up my PhD at the moment, so yeah, any questions?
Okay, we've time for some quick questions for Jessica.
So given the surprisingly high number of very closely related individuals, have you done like the back of the envelope, close kin, mark recapture, population size estimate and see what it would give you?
Yeah, I haven't been able to just yet, but I am working on it, yeah.
Well, thanks for your talk. You mentioned the Y chromosome. How long was it in comparison to X chromosome?
It's actually almost a similar size. It's only slightly smaller than the X chromosome, which is very unusual for a shark genome, yeah.
I see, interesting, thank you.
Okay, thank you, Jessica. Okay, next up is Leah Palm.
Hi, everyone. My name is Leah Palm. Thank you for joining me today when I dive into the population genetics of the Taenia lemma that we have in the Red Sea. So most of you have probably seen this stingray, right? You've seen it either walking along the shallows, in aquariums, while diving, snorkeling, maybe even in the past couple talks. This stingray is found all across the Indo-West Pacific, kind of along the coastal shallow water areas, and it is pretty common to see. So common, in fact, that it is considered a least concern species globally by the IUCN Red List. Now, when I say least concern, I mean that currently the populations are stable, but they are not actually completely unthreatened. So, like many other elasmobranch species, you will find that, you will find these individuals at fish markets, and they're often a food source. In the Red Sea, in Saudi Arabia, they're not as common in fish markets. However, we do have a different kind of threat, and that is that they are subjected to rapid coastal development. And that means that their primary key habitats, like their nursery grounds, are at risk. And this is a threat that we find commonly among many different types of rays. So, so one of the main problems that we have when we are talking about looking into a ray is that we don't have enough information about them. So that's where we implemented population genetics, which is used as a tool to measure basic characteristics of animal populations. And this understanding of a population connectivity can actually help us protect these species by understanding the gene flow and also how they will adapt to their environment. So we set out to define these genetic profile of the Red Sea lemma by assessing population genetics, the gene flow, using the CO1 region of the mitochondrial DNA, and single nucleotide polymorphisms across the whole genome. And together, these two offer us the ability to look more closely at the possible gene flow, as well as assess whether there is any potential sex bias dispersal. Now, we sampled a total of 169 Taenia lemma across northern, central, and southern Red Sea. And 126 of these samples were used for the CO1 analysis and for the mitochondrial, and then 166 of these samples were sent off to DART in Australia for SNPs sequencing. Now, most of these rays were caught by using the typical hand seine netting combination with dip nets, and we worked them up in a holding pool before we released them back where we had caught them. And from each of these rays, we took the fin clips from the trailing edge of the pectoral disc, and these samples were then processed later on in the lab. Now, from the actual processing, we found that there was a total of 11 haplotypes, which can be seen are pretty mixed across major haplotypes. And this suggested that there might be a partial genetic homogeneity. However, as you can also see, this major central haplotype is then surrounded by a couple of smaller mixed haplotypes, which is suggestive of a star-like structure, which leads us to believe that there is a recent population divergence happening. Now, what we found in the haplotype network was then backed up by our AMOVA data, and as you can see, while there was weak structure, it was technically still significant. And basically what this is telling you, this FST is telling you, is that 3.2% of the total variation was actually caused from the variation between populations as opposed to within the populations. Now, when we take a closer look at where that significance was actually coming from, it's clear that the structure, the overall significance was being primarily driven by the comparison between the southern population and the central population. Now, on its own, this could very well mean that this might be an artifact of limited data. However, it is something that is still interesting to look at, especially because this does not follow the geographic gradient that we would expect. Now, this is why we took a closer look into the SNPs, the genomic data, and when we looked into that, we actually found a much stronger structure, with 18% of the total variation occurring between the populations rather than among. Now, this might still seem like a pretty low percentage, but this is fairly normal given that we're looking at a single species. So this is actually stronger evidence of a population subdivision. And unlike the mitochondrial data, we actually have significance between all of the comparisons. So the genetic structuring between these populations was pretty strong. And on top of that, it actually followed the geographic gradient that we're seeing. So what does this actually mean? So the two marker types actually tell a consistent story, but it's just on two separate timescales. The CO1 shows us a more shallow maternal shared history, while the SNPs reveal a stronger population structure. And together, they suggest that while there is a recent shared ancestry, it has limited population genetic flow now. Now, that means that a species that we first initially thought was much more homogeneous is actually showing more population subdivision, especially following along this geographic gradient. And why do we care? I just told you that this is a least concern species. Well, even in a broadly distributed ray, this is something that we see across the Red Sea. These species are, while this species is a least concern species, we have other species that do very similar things. They are along the coast, they are seemingly not interacting as much with each other, and so the blue-spotted stingray can actually be a great way to plan other conservation efforts. So if we have this kind of structuring with this ray, what does that mean for the others? And since this is the most accessible ray that we have at the moment, it can be kind of a stepping stone to further researching other rays as well. And with that, I'd like to take the final moments to thank everyone, including my collaborators, funders, and the entire team that I had behind me. Honestly, this kind of work as all of you know, it really takes a village, and I am very proud and grateful for the village that I have with me. And yeah.
Okay, any questions for Leah?
Thanks, that was really interesting. I would just love to hear more about your conclusions about the sex-biased dispersal. Since, you know, you use one part of the mitochondria and then like some nuclear SNPs, like, what, yeah, do you think like you can rule it out completely or what?
No, that's a great question. So as of right now, the data isn't strong enough to make a proper conclusion about sex-biased dispersal. Obviously, the mitochondrial is matrilineal, so if you would kind of expect that with these kind of species, you have the females staying pretty central to where they are and have the males moving around. But that's not necessarily reflected in the data, as we have the SNPs results showing structuring and the mitochondrial data showing some structuring, but less so. So honestly, the data that we have right now is not strong enough to make a solid conclusion, but it is something that I would like to look into further.
Okay, thank you,
Potentially
Leah.
with other methods as well involved.
We'll have to stop it there. We're drifting a little. Okay, next up is Shigehiro Karaku.
Okay, my big pleasure to be a part of this session of the Sharks International. Okay, the kind of my talk, I mean, the sex that I'm talking about is not behavior, but not about population. I'm talking about sex determination mechanism. Yeah, you can probably imagine what's happened in humans or other mammals. I mean, mammalians have male heterogamety. That's determined by XY system. And then all birds are determined with the female heterogamety. That's GW system. And many fish, ray-fin fishes, have really variable systems. And many, actually, fish have sex change during lifetime. And then, as far as I know, there is no obvious documentation of shark sex change. Am I right? Yeah. So the boundary between sexes, for sharks and rays seems to be really clearly determined. That's what I'm gonna talk about today. The background of our study is this consortium. It's not really like a high throughput factory. We are really slow, but yeah, we try to produce more and more whole genome data. But we usually work for addressing biological questions, so that's why we are not really fast enough. In some of our past studies, yeah, we worked on, karyotyping, and then, use this as a kind of cross-check of the whole genome sequencing data. This is quite tricky because we need this, culture cell production for repetitive analysis of karyotyping. Another study also covered photoreception of whale shark. We didn't really sacrifice any animal. We synthesized the DNA and then let it be expressed in human culture cells, and then did photospectrometry to know what's special about whale shark vision. Our interests are really diverse, but today I talk a small fraction of our interest in sex determination. So the question with sharks and rays are usually really, easy. We can tell from external morphology, to find claspers. But things are not really, straightforward if, the claspers are not really visible. Yeah. And then genomics usually takes time, costs a lot, and do not really provide any, ready-to-use solutions. But I actually have a hope that I'm gonna mention in the end of my talk. So we don't really focus on a single species, but this is the most important species for us, that we can get fertilized eggs from aquariums. Then I don't get into the details, but sex determination is genetically encoded firmly in developmental program, and then that's decided somewhere, in early development like this. It's like a 3 centimeters embryo. And then, as I said, there is some description of 6 chromosomes. When we started our study, there were about 10 species. We'd already described, cytogenetically described 6 chromosomes. But the limitation was that it was not really linked with DNA sequences. So we couldn't really talk about the correspondence between different species. So the way how we identify 6 chromosomes were partially already described in a former speaker, but we tried to find a chromosome with a distinct coverage ratio between male and female, and then we always try to cover multiple individuals. Then, we found this a little bit distorted pattern, but only in males, which means male heterogamety. And then this actually applies to many other species that we analyzed. And there is one exception in which we identified multiple X chromosome, but that's kind of exceptional. So here is a kind of, phylogenetic tree and also the correspondence between X chromosomes. The important thing is that we looked into the lineup of genes included in this, potential X chromosome, and I found a very high, level of commonalities, high level of, cross-species sharing between these different species. So we think that the origin of the X chromosome of all of these species can be traced back to the common ancestor of sharks and rays, which is like 300 million years ago. So using this, our data and also the accumulating data from other groups, here is a kind of listing of already identified, firmly identified X and Y chromosomes. Y chromosomes are still really difficult for us. And then, this is what I mentioned. I mean, even though we cannot really tell from the morphology, from body parts or blood sample in the freezer or fins in the market, we can tell male or females by easy, a PCR marker that we designed, in this case for whale shark. And then, even if we couldn't identify the Y chromosome, we can tell male or female based on the differential dose of the X chromosome between male and female. This is the case about cloudy cat shark, that we detected the one cycle difference in the PCR amplification with the quantitative PCR experiments. This can be applied to other species. So here's a summary, and then I would like to thank all of the people who supported, contributed to this work, and then I'm happy to take questions. Thank you very much for your attention.
Okay, any questions for Shiggy? Okay, well, if there's no questions, I'd like to thank all.
There is one there.
There is one.
Sorry, there's a technical mistake. So I was wondering about, do you found any, like, what you call sex change during the development stage of the shark? That's the possible that's make up X and Y changing during the time of the development?
I said that there's no documentation of sex change in sharks and rays.
Okay, thank you.
Yeah. And then I want to add that the preprint has not been made available publicly. So it will be out soon, I must say. Thank you very much.